The arts are the salt of the world, as salt relates to food and art relates to technology. A very good morning to one and all present here. Sujit, welcome you all to the occasion of AMIFOST 2022. Today, we will be having oral presentations. I would like to invite our esteemed for the chairperson for the session. Really sorry. I would like to invite our esteemed chairperson for the session, Dr. Sujata Pandit Sharma, Assistant Professor of Department of Life Sciences at Sharda University. Dr. Sujata Pandit Sharma is presently working as an assistant professor at Department of Life Sciences, School of Basic Sciences and Research at Sharda University. She is doctorate in food science and nutrition from CSK Himachal Pradesh Agriculture University, Palampur, HP. She has more than 14 years of experience in teaching and research and guided more than 30 dissertations, MSc, MTech, and 3 PhD students. Her research is focused on development and quality evaluation of functional food products for infant nutrition. Before joining Sharda University, she has served as Associate Director with Research and Material Sciences. She also worked with FICCI and R&D head as R&D head. She published more than 25 research papers and articles in national and international reputed journals. She has also been invited as an external resource person to deliver talk on various aspects of nutrition by various autonomous bodies like FICSI, FASI, which is FSSAI, and FCI Gurgaon, and other government and private university. She is a lifetime member of various bodies like AFST Nutrition Society of India, NSI, Indian Journal of Agriculture Biochemistry, I, IJAB, and Association of Microbiologists of India, AMI, etc. She is an academic member slash reviewer of various journals. She has completed 6 to 7 consultancy projects. She has work experience with NAIP, Department of Life Sciences, national and foreign international projects. She is M-panel trainer with FOSTAC, Skill Development India, and Food Corporation of India. Welcome, ma'am, to the session.
Thank you for this introduction. Thank you so much.
Thank you. Now, I would like to invite the co-chairperson, Dr. Deepti Sharma. Dr. Deepti Sharma is a food technologist having 14 plus years of work experience, presently working as an assistant professor at Shyama Prasad Mukherjee College for Women, University of Delhi. She has also completed MBA and PG diploma course in food safety and quality management. She has attended many workshops and training programs and has presented many articles in national and international conferences. Her articles have been published in journals. She has authored 4 books, chapters in book, edited a book, and conference proceedings. She has been member of LOCF Syllabus Formation Committee of BA program, Home Science University of Delhi. She is on editorial board of various national and international journals. She is an active member of various professional bodies like AFSTI, SIB, and ISTE. She has been conferred with Young Scientist Award in Food Technology field and has received Best Oral Presentation Award in a conference. Welcome, Deepti ma'am.
Thank you.
Thank you, ma'am. Now, further moving on, I would like to invite our respected session coordinators, Dr. Neha Sharma ma'am and Dr. Lavleen Sharma ma'am.
Thank you, Sujit. Can we move forward?
Yes, ma'am, sure. Now, beginning with the technical session 6, which is food diversity and food security, I would like to invite the first group of speakers consisting of Charu Gupta and Dhan Prakash, who will be talking on the topic of phytosterols as nutraceuticals in cardiovascular aging. The rules for the presentation are as follows: that you have to speak for about 4 to 5 minutes, and the discussion period time will be about 1 to 2 minutes. You can go. Thank you.
Good morning, one and all. Am I audible?
Yes, yes, Dr. Charu, you are audible.
Okay. Hello, everyone, and a very good morning to all the esteemed guests and faculty members. Okay, I'll share. Can I share my screen?
Yes, ma'am.
Okay, is my screen visible?
Yes, it's visible, Dr. Charu.
Okay, so today myself Charu Gupta, and I am working as an associate professor in the Department of Herbal Research and Studies, and I would be presenting on the topic phytosterols for the cardiovascular aging. Now, before I come to the main topic about the phytosterols, it is an important phytochemical which is of very important nutraceutical importance. So, what is nutraceutical? It is actually the word that has been derived from the two words like nutrition and the pharmaceutical value. So, this was developed by Dr. Stephen D. Wallace in the year 1989, and it was based on a very interesting philosophy which was given by a Greek physician, Hippocrates. One should focus on the prevention, that is, the food be your medicine and the medicine be your food. So basically, the concept was that instead of using the pharmaceuticals or for the drugs in the treatment of disease, one can manage several diseases through the use of the nutraceuticals. So there are many herbs and the medicinal plants which are having very important phytochemicals like aloin in aloe vera, allicin and garlic, and so on. So they are having a very important nutraceutical value. So one has a green tea which are rich in antioxidants, and these have been particularly because of the presence of the phytochemicals like catechins, which are responsible for the decrease of the cardiovascular disease. So there are numerous areas which are covered by the nutraceuticals, but I would be dealing here for the particularly for the regulating the cholesterol lowering effect and the cardiovascular diseases. So this is because, like I told you, there are various phytochemicals which are present in the variety of the medicinal plants. Like for the diabetes, an important plant is gurma, that is Gymnema sylvester, and it is having the gymnemic acid as the major phytochemical, which is having the anti-diabetic effect. Similarly, Calmig Andrographis paniculata, its leaves are having the andrographolites, which are known to be a very good anti-febrifuge and as an immunomodulator. So without now I'll be coming to the main topic, that is the cardiovascular disease. Like before we start about the phytosterols, I would just like to focus on this graph which shows what are the prevalence of the cardiovascular diseases. So within the age group, you can see from the 20 to 80 plus years, the disease is prevalent in both the sexes, that is whether it can be a male or a female. And if we see the cardiovascular disease burden in India, one can see that the cases from the year 1990 and the death rate, they have almost doubled, more than doubled cases. So it is of great importance to deal with the cardiovascular diseases. Now, similarly, in the next diagram figure, one can see that there are various factors that are responsible for the cardiovascular deaths. So it can be tobacco, it can be low physical activity, and as well as the dietary risk. So, but the dietary risk is the most important factor that contribute to the highest rate of these cardiovascular disease death. So one can focus, use these foods and phytochemicals in lowering the blood cholesterol level. So one can have the maximum benefit without by doing certain diet modifications, because if we are consuming any chemical drugs, then obviously they are having the several side effects. So phytosterols, the concept was came as early as in the year 1950, and as the name indicates, it is basically the combination of the plant sterols, and they are basically soluble in alcohol, and they are present in the plant cell membrane as a phospholipid bilayer, and they are produced by the plants in response to the high temperature conditions. So in order to stabilize the plant cell membrane, these phytosterols, they are produced by many plants, and the most abundant phytosterols are the cytosterol, camposterol, and these phytosterols, although they are normally present in the various diet, but their amount is very less. So that is around 200 to 400 mg per day. But in order to have a therapeutic effect, our diet should have around more than 2 gram per day. So if we want intake, it has been found through the research that the daily intake of at least 1 to 3 grams can reduce the total cholesterol level by around 10%, and particularly the LDL cholesterol, that is your bad cholesterol, by around 15%. Now, if we look into the chemical structure, one can see that structure is quite similar to the cholesterol molecule, and it is having the 4 steroid skeleton A, B, C, and D, like it is visible. And this carbon 3 is attached. The atom of C3 atom is attached to the alcoholic OH group, and this, at the C17 atom of this D ring, it is attached to this aliphatic side chain. And there are basically 2 types of phytosterols. One is the sterols are having the double bond between the C5 and C6 carbon atom, and stenols, they are the saturated molecules, they are having the single bonds. Now, if we differentiate between the phyto and the cholesterol, so one can see that the phytosterols, they are helps to maintain the temperature of the plants, and they are synthesized by the plants, but the cholesterol, it helps in, they are synthesized by the liver from the acetyl coenzyme, and it serves as a precursor for the steroid hormones, bile acids, and vitamin D. And for the phytosterol, in order to have a therapeutic effect, the daily intake should be 1 to 3 grams, whereas for the cholesterol, the daily intake is recommended to be less than 200 mg, because the body can also synthesize it. Now, these doses of the phytosterols, they have been devised after numerous research studies that have been carried out, and it has been found, like one of the research study has been shared, that if we increase the dose of the sterols and the stenols to up to 2.5%, so there was a corresponding increase in the reduction. There was a corresponding reduction in the level of the low density lipoprotein cholesterol. So if we see the mechanism of the action, that how these phytosterols, they actually work. So it actually reduces the absorption of the cholesterol. Since its structure is similar to the cholesterol, so it basically competes with the cholesterol molecule into the bile salt micelles, and it helps in the interference of the absorption of the cholesterol. So less cholesterol is absorbed, therefore that results in the lower blood cholesterol level. Similarly, there is another mechanism that has been proposed, that this phytosterol, it also helps in partially soluble, precipitates the soluble cholesterol, and it converts the absorbable form into the non-absorbable form. Similarly, the phytosterols, they also enhances the cholesterol excretion into the body because there are certain transporters which are present in them. Now, besides, like this is the link which shows the mechanism, but I'll be skipping because of the shortage of time. Now, besides lowering the cholesterol level, there are other functions that has been found to be proposed, like it has the anti-carcinogenic immune regulation. It helps in reducing the prostate enlargement, and it also helps in controlling the diabetes and the inflammation. So these results has also been devised on the basis of the research studies which has been carried out. Like for the cardiovascular disease, the study was carried out with the people which are having a history of hypercholesteremia, and they were given the spreads which are fortified with around 1.8 to 2.8 grams of sterols, and they were given for the period of 1 to 3 months, and it was found that that lowered the total cholesterol level by 7 to 11%. Similarly, in the case of the enlarged prostate gland, which was a clinical trial study, was carried out for a period of 6 months, where 200 men were selected which are having the symptoms of the enlarged prostate gland, and when they were given the dose of around 60 mg per day, so it was found that they have increased the urinary flow, and there was a decrease in the residual urine volume. Similarly, for the inflammation and for the treatment of the rheumatoid arthritis, the 2 mg of sterols, when they were given per day for a period of 3 months, it helps to decrease the inflammation. And similarly for the blood sugar level also, so 2 mg of cholesterol when given for a period of 6 to 8 months once a day, so that has been found to normalize the blood sugar level. Now, in yet another study, it's a very interesting study where it was studied that the combination of these phytosterols were given along with the exercise was recommended. So the synergistic effect was obtained. Like suppose 1.8 gram per day of the plant sterols were given along with the exercise, which was recommended 3 times a week for under the supervised conditions. So it was study was carried out for a total duration of 8 weeks, and it was found that the combination is working very effectively, and it helps in reducing the level of triglycerides, and it increased the level of the good cholesterol level, that is your HDL cholesterol, and it decreased the low density lipoprotein, that is your bad cholesterol level. Now, regarding the safety considerations, now various studies have been carried out, and it has been found to be one of the most safe substances. And since it is, although there is a rare genetic condition that is phytosterolemia, which has been observed in very minute cases in 1 in 50 lakh people. So it is a type of a lipid storage disorder, but neither the phytosterols, they are neither found to be mutagenic nor teratogenic. And the levels of the blood fat soluble vitamins was also not found to
Dr.
be
Charu,
affected.
please wind it up. Your time is up.
Okay. So
Yeah.
coming to the sources of phytosterols, they are basically found in the vegetable oils and the products. So you can see rice bran, corn, flax seeds, they are full of phytosterol contents. Besides the vegetable oils, the fruits, vegetables, nuts, legumes, they are all the rich sources of the phytosterols. But since the quantity of the phytosterol is present in very minute amounts, so it is not possible to consume these lot of so many of things in great amounts. So therefore, the concept of the nutraceuticals came into play here. So nowadays, the phytosterol capsules and the tablets, they are available in the market. So that can be consumed in order to have a therapeutic effect, because from the daily diet, one cannot have the required amount of the phytosterols. And regarding this, FDA has also approved a variety of the food products where the phytosterols can be added, like margarines, your mayonnaise, pasta and noodles. And there are various, itself in India, it has also been approved as a food ingredient in the year 2018. Besides India, there are various European countries like US, Canada, Europe, where it has already been approved as a food additive. And if we talk about the stability of this phytosterol, so it was found during the processing conditions, it can tolerate the harsh conditions more than 100 degrees Celsius. It has been found to be microbiologically inert, do not contribute to any contamination, and it is also stable to a wide range of the pH. And these are the number of the various brand names where they have been producing, they have developed the food product which are fortified with the phytosterols, like yogurts, milk drinks, bread, snack bar. And the process of the extraction of phytosterols has also been developed from the vegetable oils, and a patent has already been granted. And regarding the adverse effects, although some effect has been found to be on the pregnant and the
Dr. Charu, I
breastfeeding
have to
women and
stop you.
children
I'm
under the
sorry
age
to
of the
say.
5 years.
Dr. Charu, please kindly stop. We have to proceed with the next one because we have 28 presentations and we have time limitations. Sorry, Dr. Charu, to interrupt you.
Okay.
Yeah,
So I'm
thank you.
open to the, yeah.
Yeah,
Thank you.
if there are any queries from the judges, they are requested to please ask.
Dr.
Dr.
Charu, what
Pandit.
is the, yes, ma'am. Dr. Charu, what is the concentration that we should consume to have a good effect? Is there any
Ma'am,
particular
1 to 2 grams per day. 1
How much?
to 2 grams per day. 1 to 2
One-
grams per day in order to have a therapeutic effect.
Okay, is it the standard? Or
Yeah,
is it
it's a general.
research?
Yeah, it's a standard. It has been recommended that in order to have a therapeutic effect, at least 1 to 2 grams, maximum 3 grams can be consumed. Because from a daily diet, around 140 to 150 mg per day we are consuming, but that is not sufficient to show the therapeutic effect in decreasing the blood cholesterol level.
Dr. Charu, I have one query. As
Yeah.
the present slide, you can see here the dietary guidelines. The in a previous slides, you have recommended some food which have known for the trans fats like mayonnaise. And what are your recommendations for that?
My recommendation is that one should go for the healthy foods, not the processed foods, and one should include a variety of the colored vegetables and fruits, because these are actually the natural sources. And there are various nutraceuticals which are available in the market. Like I showed various bottles also. So one can, instead of using these processed foods like mayonnaise or spreads, one can go for with that nutraceuticals supplements or dietary supplements, but under the supervision of doctor, because some studies showed some adverse reactions in the breastfeeding and the pregnant women. So studies are still going on because this phytosterol are still not prevalent much in India. The people are not generally not aware. But it is a very important phytochemical for the controlling the cardiovascular diseases. And it is having a very great potential in the coming future also.
Thank you.
Thank you, Dr. Charu.
Okay, ma'am. Thank you so much for giving
Thank
me
you.
this opportunity. Thank you.
Thank you. It's once again requested to all the participants that please stick to the timeline. It is 4 minutes for your presentation and 1 to 2 minutes for the queries. Thank you, all of you. Sujit,
Yes, ma'am.
please proceed.
Thank you, Charu ma'am, for shedding light on your topic. Now we would like to move on to the next group of presenters. We have Khushbu ma'am, Nutan ma'am, Christina ma'am, and Asha Kumari ma'am, who will be presenting the topic of comparison of extraction efficiency of different gelatin extraction method from pink perch skin and bones.
Good morning, all of you. Good morning, respected judges and my teachers and my colleagues. I am Khushbu. I'm a research scholar in Amity University, registered in Amity Institute of Food Technology. And today I will be sharing my work on my PhD thesis. Can I share my screen? Am I audible?
Yes, ma'am.
Yes.
Okay. So my work is on comparison of extraction efficiency of different gelatin extraction methods from pink perch skin and bones. Global fish, let's begin with the background, a little bit of background knowledge. So the global fish consumption or production in year 2018 as per FAO was 179 million tons, and approximately 87% of which is directly consumed for human consumption. So that leads us about 20.5 kg per capita of human consumption. So now if such a lot of human consumption, fish is being produced and consumed, that produce a large amount of rest raw material, byproducts, co-products. These are the similar terminologies. So what does this rest raw material or byproducts composed of? It is majorly composed of head, viscera, frame, skin and bones, tails, fins, etc. So if we talk about in percentage, so approximately 30 to 40% is consumed and a similar amount is being discarded as waste. And what is the current usage? Current usage is either the production of low value products or they're just sent directly to the landfills, incinerated. That is leading to an environmental hazard. However, these rest raw materials are equally important source of important biomolecules, one of which is protein. So in our studies, we have used skin and bones that is being obtained from surimi industry as a rest raw material and utilized it for the production of gelatin. So first of all, we have done the proximate analysis of the skin and bones that is obtained from the surimi industry. Majorly, it is composed of moisture and it has a good percentage of protein, up to 10%. Then we did the mineral analysis so that we can identify if there is any heavy metal present in the rest raw material. And on doing the mineral analysis, there was no heavy metal that was present. Majorly, it was composed of calcium and phosphorus only. Then we did the microbiological analysis, and microbiological analysis we conducted total plate count, yeast, mold, salmonella, and lactobacillus. And we find that no pathogenic bacteria was present in the rest raw material. Then we did screening of the gel extraction methods, and in screening we used basically two broad categories of methods. One, it was a two-step process in which treatment was conducted using sodium chloride, and another was a single-step extraction method in which the whole extraction was conducted with the help of acetic acid and water. And this is a broad scheme of what was done. The pink perch skin and bones that obtained from the surimi industry, extraction method was done. And then the gelatin extraction was obtained in the liquid form. It was lyophilized and it was a powder that was obtained. Now the process was characterized on the basis of two parameters. One is yield and another is L-hydroxyproline content. Yield is obviously expected to be very high, and L-hydroxyproline content is an indicator of the quality of the gelatin. So in this we can see that the process 3 and process 4 is that of acetic acid, the single step extraction method that was used using acetic acid. In process 3, we conducted the extraction at pH 3, and in process 4, we have conducted the extraction at pH 5. And we can see that we obtained a higher yield from process 3 and process 4. And similarly, a higher L-hydroxypolymerase content obtained from the extraction using acetic acid. That was somewhere comparable to that of commercial bovine gelatin. Then we conducted the proximate analysis of the gelatin extracted. And here also we can see that the gelatin obtained from acetic acid extraction had a higher content of protein content and a comparatively very lower content of ash content. However, that the extraction conducted using sodium chloride had a very high content of ash content and comparatively lower content of protein. Then we conducted molecular weight distribution, and in these we obtained this electrogram. In this electrogram, we can say that the extraction process had a significant effect on the molecular weight distribution. In the process 1A and 1B are the molecular ladder. 2 is process number 1. 3 is process number 2. 4 is process number 3. 5 is process number 4, and 6 is commercial bovine gelatin. And in this, we can see that as we change the extraction method, the molecular weight distribution changes. While we are talking about electrogram 2 and electrogram 3, we can see that it is corresponding to extraction using sodium chloride. And we see a wider range of molecular bands that was being obtained. This is an indicative of more degradation that has happened, more due to hydrolysis process, in comparison to extraction using acetic acid, where a higher, more density was obtained from higher than 100 kDa, which was similar to commercial bovine gelatin. So to conclude, we can see that on the basis of presence of good protein content and lack of any heavy metal or microbial contamination, pathogenic microbial contamination, pink pus skin and bones obtained from surimi industry can be utilized for development of value-added products. Utilization of acetic acid as a promising way or sustainable way for development of these byproducts into a value-added products, and single-step extraction method can be further optimized to increase the yield and hydroxypolymerase. Thank you.
Thank you, Ms. Khushbu. If there are any queries from the judges, they may please ask.
Yeah, Khushbu, can you explain a bit about your comparative study? I could not. Yeah.
Okay. Let me go back to the slide. I'll just do this again. So in my comparative study, we have chosen 2 parameters, 2 basic broad methods of comparison.
Just wait. So a sample I want to see, is there a different type of samples, or you are just working on a same sample?
No, ma'am. This simple sample. This is the pink pus skin and bone sample that we have obtained from surimi industry.
So you have compared bones and skin, right?
It is a combined biomass.
Okay.
From surimi, it is a combined biomass. They pass it through a roller, through a builder roller, so that the flesh get extracted and a combined biomass is obtained.
So comparison in between what? Comparison means what type of samples are there for the comparison?
And sample is one. Comparison is between the extraction method.
Extraction method. Okay. So what are your findings?
My findings is that the 2 methods that we have obtained, one was 2-step process and another was a single-step process. So the single-step process was providing more promising results in comparison to the 2-step process.
Okay, so you will recommend single-step process.
Yeah. Yes, I'm recommending that further optimization of the single-step process needs to be conducted. Okay. And it is a more efficient method. Since it is a single-step process, it will provide lesser constraints. It will provide easier handling, more economic, more sustainable method.
Okay, now my last question. What are the applications of this gelatin?
Ma'am, gelatin is a wide range of application, from food to medicinal industry to cosmeceuticals to everywhere. Food, we can use it in development of desserts and all sort of products that need a jelly-like consistency. And in the medicals, we can go for scaffoldings and other tissue regeneration kind of products.
So what do you call it?
Yes, ma'am.
What do you call it as an ingredient? What do you call it? If we are using,
we used to call it gelatin only. So far, we are using it from the commercial.
It will be no. It will be it will be called as additive.
Okay, yes, in food we will be calling it as additive. For example, when we are adding it in ice cream and all, we will be calling it as additive, gelling agent, that we will be calling it. Yes.
Okay, thank you.
Yeah.
Pushpa, just let me know one thing, like how can you further purify it? Or is it the purified form that you get it from this process?
Ma'am, the purification is entirely depending upon, like, for example, in this method, we can see depends upon the all the factors. Like, for example, the one we are obtaining from the acetic acid concentration is a very high percentage of protein itself. When we do this, in this process particularly, what we are obtaining is a purified form. For if we are going for another some different process, then we may get some sort of impurities, and depending upon what kind of impurities you're obtaining, then the suggested method is obtained. But in this particular extraction method, the single step extraction method, what we are gaining is a very high form of purified gelatin.
What is the percentage of purity?
Ma'am, the percentage of purity, ma'am, we haven't yet conducted the percentage of purity. We had determined the purity on the basis of the ash content. The ash content was among the limit of what was prescribed by FSSAI. That was less than 4%, I guess. And on the basis of that, the parameter that was already prescribed, we have determined that it was edible. It was of edible grade quality that we have obtained. And further, the results were justified using the SGS page analysis. Had it been there a different form of impurities, we would have obtained it in different bands in the SGS page.
Can this gelatin be used in vegetarian preparations?
No, ma'am, not at all. This will be strictly used for non-vegetarian preparations.
Okay, fine. Neha ma'am?
Yes.
Next candidate.
Thank you. Thank you, Pushpa. Suji?
Yes, ma'am. Now, moving on to the next group of speakers, I would like to invite H.C. Devaraj, Monica Sharma, K. Jayaraj Rao, and M. Anupama to present on the topic Nutri-Cereals Incorporated Probiotic Dairy Spread, Development, and its characterization.
Yeah, thank you very much. Am I audible, madam?
Yes, yes, Mr. Devraj. You are
Yeah,
audible.
thank you, madam. Let me share. Is my slides are visible?
Yes,
and
yes,
it's
Dr.
moving?
Devraj. Yes, Dr. Devraj.
Well, thank you very much, madam. Respected Amity family and respected judges, dear participants, ladies and gentlemen, good morning. I am Dr. Devraj, scientist from ICR, National Dairy Research Institute. I'm presenting my research topic entitled Nutri-Cereal Incorporated Probiotic Dairy Spread, and we'll be discussing development and characterization. As our Amity founder chairman, Dr. Ashok K. Chauhan sir, rightly pointed out in the inaugural session that India could become the world leader in promoting cereal or millets to the globe. And to support that, India, of course, is the major producer in the globe of cereals, millets. And because of India's effort, so 2023, we are celebrating as an International Year of Millet. And India in 2018 also, it's in a gazette, it's notified, India brought these millets under the umbrella of Nutri-Cereals because of its food and nutritional security and sustainability to the harsh climate conditions. So in that sense, Nutri-Cereals having excellent prebiotic and added health benefits, so it could be one of the good source for developing functional foods. And as we know, this changing lifestyle and increasing work population, people are looking for convenience, ready to eat, and functional foods. So milk, of course, is a good in itself. It's a complete food, but still lacks in fiber and some of the micronutrients, which could be easily complemented with millets. In that way, milk-millet composite is an excellent matrix for the development of probiotic, its development, multiplication, and survival. With this concept, we are working on the development of composite probiotic dairy foods to cater to the needs of health needs of the fast-growing consumers. So basically what we did, we checked the prebiotic potential of finger millet and sorghum, and you could see in the graph that the prebiotic potential, of course, the finger millet and sorghum increase the viable probiotic count to the tune of 1 to 2 log compared to control. So this is an excellent prebiotic potential of the finger millets, and it is consistent with the selected strain like L. Elastophilus, L. casei, L. plantarum. In all the 3 strains, it given the consistent result of increased viable probiotic count. Of course, you can visualize in the picture also here, it's a higher count for the sorghum enriched compared to skim milk base. Well, LKC and L. plantarum consistently given good result even to the tune of 10 raised power of 10, 10, 11. So in that sense, LKC and L. plantarum, we took it forward for the development of the product. And to support the same, the acidification rate of the mixture, which is with the skim milk, 24-hour germinated sorghum, LKC and L. plantarum, so produce excellent acidification rate, indicating that the LKC and L. plantarum strains that we have selected, so having symbiosis between them and their growth rate. And to the continuing the same, the germination of sorghum and millets, germination has got significant effect on the viable probiotic count, and added to it, it reduces the anti-nutrients like phytates and also significantly improves the flavor of the final product. In that way, so sorghum and millet flour germinated to 24 hours is further taken for the product development with LKCI and L. plantarum are the probiotic strains. And then we standardize the product using optimal mixture design technique based on our preliminary studies and also experience in the field. So we listed some variable components and fixed components. Among the variable components, probiotic curd, butter, finger millet or malt, sorghum malt, milk protein concentrate to increase the protein content, and stabilizer, sodium tripolyphosphate was used in the range selected based on the preliminary studies. This is the flow diagram where fat phase and water phase, the optimized ingredients of the water phase and fat phase are mixed, blended, heat processed, and then homogenized. During homogenization, the probiotic curd prepared was added, and then it is mixed in the high shear blenders or mixtures we can use to mix them and to produce probiotic dairy spread. And this is the design suggested 25 runs where we did a sensory and textural parameters. We load the values to the software, and then finally after some statistical analysis, and also software suggested the optimized formulation, and we use this formulation for preparation of the final product. And then we packed it in the convenient packages for use, for convenience use, in like stand-up pouch with spout, also in the flexi tubes. So both are running fantastically fine. And this is a product optimized, the formula optimized for the preparation of the probiotic spread, and which contained, the final product contained 10 raised power of 3 log CFU per gram, the viable probiotic count. So that was really good for probiotic. And of course, it contains major like total solids, like especially protein, almost 16% protein. So that's good in terms of the protein. So to conclude the topic, the sorghum and finger millet exhibited excellent probiotic potential, and the mixed strain like LK indicating the symbiosis of the product. In the future course of time, the composite dairy drink, yogurt, dahi, like cereal based or millet based products we can able to produce. Like this particular product, we are under the technology has been already transferred to one of the dairy startup, and also paperwork is undergoing to transfer the same technology to Karnataka Milk Federation as well. So these are the two different technologies, and again added to it some other technology like dips also we produced. This is finger millet based, this is sorghum based, and same finger millet and sorghum based dips also we are in the process of production. Thank you very much. Let's all join our hands together to promote the millets consumption and for the welfare of the human race as a whole. Thank you very much, madam, for giving this opportunity.
Thank you, Dr. Devraj. Dr. Pandit and Dr. Sharma, if there are any questions.
So
Very
one
good.
thing like, yes ma'am, आप continue करिए. सुजाता मैम आप पूछ रही थी फिर मैं पूछती हूँ.
Ma'am, I think she is on phone, तो आप carry out कर लीजिए दीदी मैम.
Sir,
Hello.
आप ये
Yeah.
बताइए, like you have used stabilizer, right? Which stabilizer have you used for making curd?
Yeah, madam. That curd actually to the product preparation, dairy spread, we use sodium tripolyphosphates as a stabilizing salt.
And did you add any other stabilizer?
No, madam,
And was
not
you able
required.
to- Okay, okay. And can we make a probiotic drink from this? Like you made a prebiotic.
Yeah, not from this formulation, but we can really prepare a good probiotic drink with cereal or milk millet based probiotic drink is, yeah, we can produce.
Using
Very-
the same culture or something different? Or you are not planning to do that?
Madam, yeah, same culture can be used. Right now, we are into the products like dairy dips and dairy spreads, probiotic dairy spreads and dips with finger millet and sorghum individually and also in combination, madam. So just to have, because this finger millet as a whole, it produces slightly brownish color. Some appearance is not so well, but of course, as a functional food, it can be accepted.
Okay, sir.
Nice attempt, sir. Your product is really nice.
Thank you, madam.
And you are promoting millets. That's also very good. My question is, why you are working only 2 millets in this?
Yeah, definitely, madam. So being a major millets, we have taken these two, we are in southern India. So we are into the southern India. So we are getting plenty of this, but definitely the minor millets works much better, I guess, because the popping, germination of this minor millets gives better flavor over the sorghum and finger millet. I definitely that minor millets works much better, what I feel.
Okay, so what is the shelf life of this dip?
Madam, dip still we have to see, but as a spread of this product, so it stay for 2 months, madam, at refrigeration temperature.
Okay, refrigeration temperature.
At refrigeration temperature, 2 months.
Okay, and what are the calorie count of this per 100 gram? You have not mentioned that in your slides.
Yeah, it
You
should be
have-
calculated, madam. Yes, really, it's not.
Yes, skip that. You have skipped that as a food because you are using so many ingredients as well as fat you are using. So that's a concern.
True, madam. But the spreads in the market actually having higher fat content than the protein content. We are trying to make it of the protein a bit higher and bring down the fat to the lower side. The spreads available in the market are having more than 60% fat in it.
I know that, but here I just asked you that that should be mentioned in your slides.
True, true. Yeah, true, madam. Definitely.
Thank you so much. Thank
Yeah,
you.
madam.
Yeah, please call next. Yeah, thank you.
Thank you, ma'am. Thank you, sir. I'll priorly apologize for not being able to pronounce anybody's name properly. I would like to invite the next group of presenters, which are S.R. Dandare, K.S. Chute, S.V. Karat Bhajne, R.B. Wagmar, and J.D. Ugade, who will be presenting on the topic of processing and functional properties of edible insects, risk and benefits associated with its consumption.
Good morning, everyone. Good morning, respected teachers and my friends. I am Sagar Ramdas Dandare. I am currently pursuing food technology in prestigious LIT Nagpur, and our topic for paper presentation or review paper is processing and health properties of edible insect and risk and benefit associated with its consumption. I did this work with my one of the colleague, Kaustubh Chote.
Good morning, everyone. My name is Kaustubh Sanjay today, and I am currently pursuing BTech in Technology from the prestigious Institute of Technology, Nagpur. So the first topic for our presentation is why there is need of other source of food. So why there is need of other source of
Excuse me,
food and
please log out from other device.
Dr.
Excuse me, you logged in.
Miss,
Sir, your voice is echoing, sir.
okay.
if you have logged in from two sets, then please log out from one. Otherwise, it will keep on echoing. Or if there is somebody else also who has joined,
Ma'am,
now it's clear.
okay. And the first topic is why there is need of other source of food. Why edible insects only? As population is increasing gradually, the global challenge of food insecurity is emerging out there. Earth's population is set to reach 9 billion by 2050. So the experts are worried about production of edible protein to feed all of us. Therefore, there is need of alternative food sources to replace traditional and less sustainable ingredients. Evidence suggests that more farming and consumption of limited natural sources will not be enough to feed all of the population of world. There are several changing environmental conditions out there, like food production causing more environmental pollution, due to which that is causing acid rain and changing climatic situations. Livestock rearing, especially beef rearing, is causing large number of greenhouse gases effect, such as methane, which is 3,000 times more, which is 3,000 times fewer greenhouse gases are emitted from the insect production, and also increasing pollution. Production of food causing more number of pollution, which is causing bad effect on waste management. Hence, by knowing all these factors, we need another source of food.
So, ma'am, due to this edible insect comes in limelight, and the term entomology introduced, that means practice of eating insects. And the key factor for human consumption of insects, their high protein content, and it can be used as a food additive, add snacks or powder from after processing, and it helps to increase the nutrient value of regular diet.
Now, why there is need of farming of edible insects? So there is variety of insects which are edible. There are around 2,000 species of insects which are edible out there. So about 92% of edible insects are harvested from wild forest. So all edible insects are not available every time in the wildlife or wild forest due to several environmental factors. The quality and safety of harvested insects of wild cannot be guaranteed. This can cause consumers to death also. And harvesting of insects from wild can cause extinction of species if we are consuming insects which are less in quantity. Then, so continuous farming of edible insects is a necessary thing to do.
Yes, ma'am. So different farming techniques used for different species of edible insects, and every edible insect can form artificially. And for farming, there are two approaches used generally. So insect can be fully domesticated and raised in captivity by simple method, and another approach is to partially raise in captivity to provide them their suitable environment for growing them.
Then, what is the processing of edible insects then? So different insects have different processing methods. Not every insect has the same processing method. They can be done by every other method. So edible insects can contain a number of nutrients that are very important in healthy living life. And to obtain these nutrients, we have to do processing on the insects. They are like frying, boiling, roasting, and others. So first of all, what we do is we remove the waste parts out of them, and then we do processing methods like roasting, frying, and boiling. Then these processing techniques help those who generally avoid insect eating practices due to disgust factor in them, for those who uh, consider insect eating as a taboo.
So, ma'am, generally edible insect shows different properties for different processing techniques.
Mr. Sagar, you have just sorry to disturb, but you have just one more minute to wind up.
Okay, sorry, ma'am.
Yeah, please
So, there are
wind up.
okay. Okay. So, ma'am, there are 6 functional properties of edible insect. First is solubility, then foaming property, then emulsifying property, then gelling property, water holding property, and oil holding property. Then the first property is solubility. So, ma'am, the proportion of protein that are in the soluble state under the specific condition is referred to as protein solubility. So, generally, solubility of insect protein is depend on the pH value, and the solubility of highly affected by processing treatment. And mainly solubility is highly affected by the type which we are using for the processing treatment. After that, there is the emulsifying property. Emulsification is known as the homogeneous mixture of two immiscible liquid, and whereas the smaller proteins are conductive to diffusion and generally show better emulsifying capacity but lower emulsion stability, whereas the larger protein have lower diffusion rate, which increase the emulsion stability after the formation of the layer around the oil and water interface. After that, there is a foaming property. Foam is generally known as a two-phase colloidal system, and the foam formation at air-water interface is controlled by the transfer, penetration, and rearrangement of molecule. And the higher foaming capacity was noted in the protein preparation. After that, there is a gelling property. Gelling property generally known as the protein ability to combine and form a gel. And generally, protein concentration, pH, ionic strength, processing, and protein denaturation affect the gel formation property. After that, there is a water holding capacity. Generally, water holding capacity, we say that the ability
Mr.
of...
Sagar, your time is up. You kindly come to the conclusion part, please, to the last slide.
Okay, ma'am. Okay,
Yeah, your time is already up.
sorry. So, ma'am, after that, there is a water holding capacity, and
Please
after that,
don't
there is the oil holding capacity.
explain. Yeah, just conclude it, because we are already running behind the schedule, and we have already announced that every speaker will be given 4 minutes and 1 minute are for queries. So that makes up your 5 minutes. You have already taken 7 minutes. So it's my very humble request to kindly conclude.
Yes, ma'am. And going on the conclusion, let us discuss some benefits of consuming edible insects. Edible insects are rich in nutrition. There is reduction in greenhouse gas emission, less feed and water required. They are fast growing. They are more economical in production. They require less land for growing, and they can be a stable income for farmers. Thank you, ma'am.
Thank you so much. Judges, if there are any questions, please.
Thank you so much. You have not given any risk factor of this. We cannot consume every insect. So there are several type of, anti-nutrient factors associated with them. You have not given that. We have not mentioned the particular type of insects which can be consumed.
Ma'am, there are several insects like
Several.
mealworm.
So you have to mention their name. Na? Several, we cannot consume everything.
Yes, ma'am.
Okay,
And
thank you. Thank you from my side, ma'am. Anyone else ask?
Thank you.
Dr. Deepti, Dr. Lavleen, any questions? Okay, so we can move on to the next participant. Thank you, Mr. Sagar.
Thank you, sir. Thank you.
Thank you.
Moving on to the next group of presenters, I would like to invite Mayuri Rastogi and Salim Siddiqui, who will be presenting on the topic of food fermentation an approach to enrich water-soluble vitamins content.
Good morning, everyone. Just give me 2 minutes to share my slide. Is it visible to all?
Yes, it's visible and you are audible.
Yes, ma'am. So good morning, everyone. My presentation topic is food fermentation, an approach to
It is
enrich.
requested to the speaker that they please switch on their cameras.
Ma'am, there is some issue in the camera today in the laptop.
Okay, please carry.
My presentation topic is the food fermentation. It is an approach to enrich water-soluble vitamins content in the food. This is my review topic, and my research work will be done on the same. So if you talk about the introduction, the fermentation in food processing is the process of converting carbohydrate to alcohol or the organic acids into using the microorganisms like yeast or bacteria under the anaerobic conditions. Or we can say that it is in any metabolic processes that releases energy from the sugar or any other organic molecule, which does not require the oxygen or the electron transport system for the final production of food. So based on the respiration, the fermentation is based on the two types. One is the aerobic respiration and the second one is the anaerobic respiration. So aerobic fermentation is the fermentation where the metabolic processes is done by the cells, metabolized sugar via the fermentation in the presence of oxygen and occurs through the repression of normal respiratory metabolism. Basically, it is called the respiration other than the fermentation. So yeast requires the oxygen for a number of processes essential for the reproduction. So wine, beer, acetic acid, vinegar are used the oxygen in the primary or the first stage of fermentation, and the exposing the surface of the vinegar to the more of the oxygen. It creates more healthy and a flavorful vinegar and control the pH. Anaerobic fermentation. The anaerobic fermentation is a method where the cells use the energy, or they liberate the energy from the carbohydrate when the oxygen or the other electron acceptors are not available in the surrounding environment. So through this process, the NADH is reduced to the NAD plus nicotinamide adenine dinucleotide, and that provides the energy for the bacterial multiplication. So there are two types of anaerobic fermentation. One is the ethanol fermentation and the second one is the lactic acid fermentation. In the ethanol fermentation, this fermentation process is used for the production of majorly the wine, beer, and the breads, and where the ethanol fermentation converts two pyruvate molecules, the products of the glycolysis, to two molecules of ethanol and two molecules of the carbon dioxide. The reaction is a two-step process where the glucose is first converted into the pyruvate in the aerobic condition, and then the pyruvate is converted into the ethanol along with the carbon dioxide. So for the fermentation, the bacteria or the yeast requires the substrates that can be included the honey, cereal grains, fruit juices, grain malt, or any of the sugars that can be converted into the ethanol. The second one is the lactic acid fermentation. So there are two major types of the lactic acid fermentation. The one is the homolactic and the second one is the heterolactic. The difference between the two lies is in the heterolactic fermentation, there is a liberation of gases like carbon dioxide, and the homolactic fermentation, the NADH reduces pyruvate directly to form the lactate, and there is no release of the gases. So this is the flow chart or the diagram of the anaerobic respiration. The first glycolysis is converted into glucose is converted into the pyruvate in the presence of oxygen, and then the pyruvate is converted into the ethanol, two molecules of ethanol with the liberation of carbon dioxide. That is that we call the fermentation, anaerobic fermentation. And for the lactate formation, the NADH is reduced to NAD, and that provides the energy for this process. So this is the systematic representation of some common types of fermentation that is used in the foodstuffs. So there are 4 types of fermentation that is very common. The one is the alcoholic fermentation, which is done by the yeast, and they use for the alcoholic beverages. Lactic acid fermentation is done by the lactic acid bacteria, lactobacillus, and the end product is the lactic acid. The acetic acid fermentation, the acetic acid bacteria, and then the final product is acetic acid. And the alkaline fermentation, which is mainly conducted by the bacillus species, and which produces the amino acids. So there are many microorganisms are used in the fermentation, but most widely used.
Ms. Mayuri, your time is up. Kindly wind up in next one.
Yes, ma'am. Just give me 1 more minute. So the microorganisms, the major found microorganisms are yeast, lactococcus, bifidobacterium, and the molds and lactobacillus. These are the common types of the microorganisms that is being used in various types of studies previously in different food groups like cereals, vegetables, dairies, and the meat products. So these are the microorganisms that is majorly used: lactic acid bacteria, fungi, and the yeast and propionibacterium. So based on the classification, the classification is majorly based on the food groups, and the other classification include the foodstuffs which contains the viable microorganisms like yogurt and yeast, and the foodstuffs which requires the fermentation during the process, like soya sauce, bread, and beer and wine. So there are a few of the advantages of the fermented foods, like it helps in the vitamin enhancement, it increases the antioxidant activity, it releases the enzyme production, peptide production, and also it degrades the anti-nutritive factors into the foodstuffs. So this is the major the benefits of the dairy products for the fermentation. Like it increases the lactose hydrolysis, it increases the lipolysis, it enhances the mineral content, it provides the antibacterial activities, and increases the B vitamins in the food product. This is the benefits of the meat fermentation, like it reduces the nitrate and nitrites. It stabilizes the
Ms. Mayuri, we will not be able to give you any more time. I'm sorry to say.
Sure, ma'am.
Yeah.
This is the conclusion now. So in the concluded, I can say that the fermented foods are being used from the previous many years, and these foods may be prepared from the grains, legumes, roots, and vegetables, fish, and meat. But through fermentation, the food can be more nutritious, digestible, and safe for the consumers. Thank you.
Any
Mayuri,
query?
what is the novel in your work? I did not find any novelty in your work.
Ms. Mayuri, are you there?
Yes, ma'am. Yes, ma'am. Ma'am, this is just a review, and we will do my research work on the fermentation. So it is just a review.
No, but ferments are practice, right? It's an age-old practice that we are following in India or in Asian countries. So, what is the new thing that you are going to add to the knowledge?
Ma'am, I have just compiled the previous studies till now, so I have presented it here, and we will use this information for the production of something new in my work.
Okay. Thank you, Ms. Mayuri.
Thank you, ma'am.
Thank you, ma'am. Now moving on to the next session of technologies for food safety and quality analysis, I would like to invite Arjun Sharma, Praveen Dahiya, and Sumedha Mohan, who will be presenting on the topic of role of plant extract to control post-harvest food wastage caused by pathogens.
Are my slides visible?
Yes, sir. Your slides are visible.
Okay. Good morning to the respected juries and everyone. Thanks for giving me the opportunity to present the topic, role of plant extracts to control post-harvest food wastage caused by pathogens under the guidance of Dr. Sumida Mohan and under the co-guidance of Dr. Praveen Daya ma'am, Amity Institute of Biotechnology. Well, coming to the introduction of my topic, the rise of population globally has resulted in increase in demand for food, even though the production capacity of our country is still high, but a lot of food is being wasted due to the spoilage, and one of the such primitive reasons are microorganisms, spoilage by the microorganisms. A report issued by the UN and Agriculture has stated that 1/3 of the food produced for human consumption is either spoiled or wasted. Human economical losses faced by both the producers as well as consumers due to food spoilage issue. On an average, nearly 470 million small-scale farmers lose 15% of their GDP in developing countries. Food spoilage is a very complex problem and the whole world is affected with it. Well, this slide represents the world top cultural countries. India is amongst
Mr.
the top
Arjun,
10.
your slides are not moving and it's not on full screen mode.
Ma'am, but I've already set it in the slideshow. In my screen, it is showing in slide.
This is what I'm telling you, that what is being coming here on the screen, Arjun.
Is it?
And yours,
Is it-
and it's, yeah, only your first slide is visible to us where you have mentioned the name of your topic.
Is it visible, ma'am?
It's
No,
still the
audio.
first slide. No, it's still the first slide.
Is it visible now?
It's the first slide. Can you reshare? Yeah, now
Is
the
it visible?
second slide. Now,
Okay,
yeah.
okay. So, ma'am, as the time is limited, can I just,
Yeah, please carry.
Okay. So coming on this slide represents the world top agriculture countries, and India is amongst the second in the top 10 countries. It's in the 5th position. According to Somadatta Report 2021, it is a huge matter of proud for us that India is amongst the top 10 countries. But when I shift to this slide, next slide, which shows the enormous scale of global food waste. It is the report which is published by Global Food Waste Index Report 2021. It says that India is at the 2nd position. It is approximately contributing nearly 6 lakh tons of waste per annum. It is a huge matter of concern that 6 lakh tons per annum food is being spoiled. And one such reason of the spoilage are the microorganisms. As we see in this slide, microorganisms can reach the food product in any of the food production stages, from cultivation through transportation till it reaches to the consumers. So this slide represents some primitive microorganisms which are responsible for the food spoilage, like E. coli affects meat products, causes vomiting, etc. Listeria monocytosis affects raw food materials, listeriosis. Norovirus affects fresh food, causes neurological damage. Clostridium affects poultry items, causes diarrhea. Similarly, Trichinella affects raw uncooked meat and causes fever, so many diseases, etc. So to remove these microorganisms, to inhibit the microorganisms, the conventional methods which are being followed by the industries, like the use of synthetic additives, are not only expensive, but they do have potential side effects on environment as well as human health. So there are some several preservatives. These are some specific preservatives which are being followed up by the industries on day-to-day basis, and there are side effects mentioned of those preservatives, which is impacting the human health, as well as like sodium benzoate, which is used in carbonated drinks, is suspected to be a carcinogen. Similarly, sulfur dioxide, which is used in potato products, it causes diarrhea. Aspartame, which is used as a sweetening agent, it may cause neurological damage. Potassium nitrate, which is used in canned meat products, may combine with other substances to form nitro, so means that are carcinogens. Calcium benzoate, which are used in low sugar products, may temporarily inhibit digestive enzyme functions. So this chart shows that the synthetic additives, like the conventional method which are being followed up by the industries, are not as far effective for both the environment as well as for humans. So therefore, we need to find an alternative approach. And plants may act as a best approach for this, because plant can possesses both the antimicrobial property by which we can inhibit the pathogens, as well as antioxidant property by which we can preserve them. So these are some plant extract which possesses antimicrobial as well as antioxidant properties. They have already been identified, like oregano, rosemary, thyme, laurel, sage, clove, basil.
Arjun, sorry
Yes, ma'am.
for interruption, but now you have only 1 minute.
Okay, ma'am. No worries. These are the specific compounds in plants which possesses antimicrobial property, like oregano, that have thymolol, rosemary has terpenoids, thyme has terpenoids, sage has flavonoids, basil has terpenoids. So these the plant extracts which possesses antimicrobial properties do have the potentials for the production of potents. It can be used in the form of spray to increase the shelf life of the food products. Already so many research work has been done on this, but it needs to be implemented more. Previously, the interest in biofilms was specific to the research related to water distribution system, but there are prominently more ways to use it. So these are some specific biofilms which is being identified by the researchers, like gore plant, gore gum, mimosa arabic gum, maize, yam, soybean, soya protein. These are the biofilms which are actually identified by the scientists, and these have they have the properties as well by which we can implement in the ground level. Then coming to my conclusion,
Arjun, your time is
coming to the conclusion, food spoilage and waste is a very critical issue, as we all know about it. The conventional method which is being followed by the industries are not providing very good outcomes. They are far very expensive and also causes adulteration to food products. And there is a demand to explore an eco-friendly alternative approach. And for that, biofilms produced using plant extracts do have the possibility for this purpose. Although a lot of research work has been contributed by our researchers, but still this technology of using plants needs to be implemented in ground level. So this needs to be further explored, as it can be served as an opportunity to reduce the food waste, improve the shelf life, and increase the nutritional quality. It is safer, cost effective, and natural as well. Thank you very much.
Any questions?
A nice presentation.
Thank you, ma'am.
Yeah, so have you worked on some edible film?
Ma'am, sorry, please repeat the question. Have I worked?
Have you worked on some edible film like you have mentioned here?
Ma'am, currently actually we are in a process of identifying some novel the plants which are less explored or something like this. Like in this slide you can see these are the plants which are being recently identified in 2021 only. So that's what I've said, that there are so many things which have been identified, but they are not implementing on ground level. Still we are using the same preservatives.
Yeah. So it's a novel technique if you will work on that. It can replace some plastic and some other packaging material. So that would be very nice.
Yes, ma'am.
Thank you so much. Yeah.
Thank you, ma'am. Thank you, ma'am.
Is this a review article or you have started working on this?
Ma'am, please repeat the question. Is this?
You have made a review article from the
ma'am, currently
you are planning to
ma'am, currently it is a part of my research. At the end I will be looking forward to create something like this. Initially it is a review only, but yeah, it is a part of my research, and we are looking forward to identify some of this. Hello, am I audible?
Sujata ma'am, yes, you are. Yeah. Thank you so much. Next, please. Next.
Thank you, ma'am. Thank you, ma'am. Thank you, Arjun. I would like to request everyone, whosoever is having a technical problem in presenting the presentation, can send their PPTs or presentations to the mail ID mentioned in the chat box. Moving on, I would like to call the next presenters, Vaishali, Harsh Sehgal, and Dr. Vandana Singh, who will present on the topic of lead toxicity and its adverse effects by food packaging on human health.
Good morning, all of you. I'll just share my screen. Is it visible?
Yes, it is visible.
Okay, thank you, ma'am. Good morning, everyone present here. My name is Vaishali, and I am a PhD scholar in Department of Forensic Science, Sharda University. And today I'm going to present my presentation on lead toxicity and its adverse effects on by food packaging on human health. So as we all know that food is adulterated by numerous adulterants like flavors, colors, preservatives, additives, and it is for the purpose of increasing the presentation of the food and the shelf life of the food. But as we're talking about heavy metals, the food is not contaminated, adulterated purposely by heavy metals, but during the process of manufacturing and processing, packaging processes, the heavy metals get incorporated in the food. After packaging, the food is in contact with the plastic, paper, or glass for a certain amount of time that leaches out the heavy metal into the food present in the packaged material. So these diagram shows how the migration of chemicals like ink, packaging, and gas content gets migrated into food through higher temperatures and long storage of time. And this slide shows how the heavy metals like lead, etc., are enters in the food chain, like the milk we drink in India on daily basis. Most of the population drinks for have milk by this packaged milk, and the drinking water bottles like Bisleri, Kindle, etc. These are directly consumed by the humans, by the infants, the children, and the plants, that leafy plants are also consumed directly by the human, and it enters the tissue system and it causes adverse effects and toxic effects, toxic deposition in the human tissues that causes toxic effects. So this is one of the source of lead contamination. Plastic, paper boards, and glass. These are the material that are frequently used in packaging food material, and by these toxic metals like lead leaches out from the packaged materials, and the color additives, adhesives that are used. The second source would be the soil that is heavily flushed with heavy metals that from the irrigated water and the pesticides, fertilizers that are used extensively, and the consumption of these leafy vegetables by human beings are direct source of lead intake. The second source will be the air, by the exertion of the industrial hazardous waste and the pollution by the vehicles and the boards, the trains, the rural practices, the industrial practices in which burning of coals and woods are used, that enters through the respiratory system and metabolizes in the tissue and causes toxic effects. The fourth source.
Vishali,
Yes, ma'am.
sorry to interrupt. You have only 1 minute to conclude your presentation.
Okay, ma'am. Okay, ma'am. The water also is contaminated by many practices, by industrial waste and municipal runoffs. The groundwater is contaminated. This diagram shows the adverse effects on human health. It interferes the infant mental development that causes low IQ, the menstrual disorder that is very common in today's world among women, that is PCOS and PCOD. It can be reason of lead accumulation. The elevated lead levels in pregnant women can lead to premature birth, and on molecular level, vitamin D synthesis and DNA replication is also interrupted. The sum of the sustainable steps can be look after, like the common agricultural practices such as the use of pesticides and fertilizers can be converted and washed over into biodegradable exercises, use of green food technology, the use of solar energy in place of coal and petroleum, hydroponics that requires no soil but only water, natural light, and nutrients. Also, packaging of food materials can be replaced, like active packaging, intelligent packaging, bioactive packaging, and etc. Nanotechnology packaging is very promising in food packaging area, and other many small limitations we can use in children and in adults. Um, and this is the conclusion that water, air, soil, plastic are the main source of lead accumulation, which end up entering the food chain, and it is consumed by the humans. And use of plastic in packaging shall be diminished over time. Active and innovative packaging should be used. Thank you.
Okay, any questions?
Yes, Vishali, you have talked about only lead. Why
Yes,
don't
ma'am.
you have considered some other parameters which are associated with this?
Ma'am, there are many other also, but in food packaging, and mostly lead is studied over time mostly, and it causes most adverse effects. And in my research, in my planning, I have planned to choose this metal. That is why I use this.
Any particular kind of packaging material is more means you have found it more contaminated with lead or some other?
Ma'am, I have not studied this. This is a review study for now. I'm planning to study further.
Okay.
Thank you, ma'am.
Thank you, ma'am. Now, I would like to hand it over to my colleague Janvi to carry forward the session. Thank you.
Thank you.
Thank you. Now I would like to welcome the next group of presenters, Twinkle Sachin, Jyoti Nishad, Chandana Sri Sai Nadimanti, Sanjana Soni, and Priya Darshini for their oral presentation of their title of abstract, Food Safety Challenges in India, Indian Street Foods.
Good morning, everyone. Let me please share my screen in just a minute. Am I audible?
Yes.
You are.
Yeah, thank you. Good morning, everyone. I am Chandana Sri Sai from Food Technology Department of Shahid Rashtriya College of Applied Sciences, Pune. On behalf of my team, I'm here to present our study on food safety challenges in Indian street foods. Our country is known for its vast food diversity, reflecting our tradition and culture in an endless variety. The cuisine of place speaks volumes about the weather, culture, lifestyle, and habits of the people. India has hundreds of street foods with their diverse ingredients and aroma across the different states and regions. On the other hand, we definitely know the challenges and how unsafe is our Indian street food. However, there are varieties of street foods with different preparation methods and many varieties. Street foods are a source of affordable, convenient, and often nutritious for urban and rural poor. It is also a source of attractive foods for tourists. Majorly, it also provides livelihood. But despite all of this, it is evident that street foods has many food safety challenges to be addressed. Street foods can be defined as ready-to-eat foods and beverages prepared and sold by vendors and hawkers, especially in the streets or other similar public places. The rapid urbanization has led to a sudden and unprecedented urban growth, marking difference in the lifestyle of the people. leading to the increased demand for ready-made foods or street foods. And consuming street foods is perceived to be a major public health risk as it might cause potential health hazards like E. coli, etc. Factors responsible for safety in street foods are lack of basic infrastructure, service, operations, and management, insufficient resources for inspection of microbiological status of the food, and also inadequate knowledge of basic food safety measures and public awareness. Significance of street food safety in public health. Uh, Street foods are consumed in wide range of population, and the low cost and accessibility and convenience are the key factors for the growing popularity of street foods. But to achieve this, the safety standards are often compromised, leading to risk in public health. This directly affects the health and nutrition of the consumers, as the vendors might use poor quality raw materials, cutlery, poor sanitary practices, and many more. Hence, it is evident that unsafe street food is affecting the health of large populations. Few most important food safety challenges in street foods are discussed here. Uh, the raw material itself is the most common source of contamination. Vendors prefer lower grade of raw materials for low cost, and these ingredients can cause potentially harmful microorganisms and toxins. Vendors should also pay attention to the water supply. Utensils and equipments and other containers should be made up of materials which do not release toxics and hazardous metals like copper, lead, cadmium, etc. into food and beverages, especially when the foods are acidic. The design, construction, and maintenance of equipment are also important to food safety. Food preparation and processing is a critical area. Foods that are eaten should be washed sufficiently with safe water to reduce contamination on the surface. Food handling and storage food handling, storage, and waste disposal practices should also be accounted. Uh, and waste products and leftovers should be disposed of hygienically. If not, a food safety threat can be posed. Personal hygiene can be a source of food contamination. So it is suggested for the vendors to maintain a proper personal hygiene to avoid contamination. Following are the control measures and initiatives to improve safety of street food. Firstly, street food management can be improved by educating the food handlers, improving the environmental conditions, and providing essential services to the vendors to ensure safety of the commodities. Secondly, enabling official recognition and control of the street food industry as an integral part of the food supply. FAO has implemented and supported several projects which aimed at improving various aspects of this street food sector, and India is one of the countries. Lastly, initiatives have to be taken to improve food safety in street foods. Considering that street foods have a significant amount of influence on public health, sanitation, and food safety, here are few regulations provided by the governing bodies. Clean Street Food is a project by FSI to ensure health, hygiene, safety standards of street foods, and also it concentrates on social economical betterment of street vendors. FSI 2011, a national policy of urban street vendors, and a scheme of accreditation of quality of street foods by Ministry of Food Processing Industries by Government of India are the other initiatives, and I'm just skipping it because of lack of time. Apologies for that. Street foods were affected during COVID-19 pandemic. Though there was no evidence of COVID-19 transmission through food, awareness increased among both the vendors and consumers. FDA also proposed the guidelines, and vendors completed digital training on COVID-19 food safety guidelines, and sanitary facilities and cleanliness were increased. I would like to conclude our study by saying that the street food safety is a process that will be attained with continuous awareness and among vendors and consumers. Food safety legislation should accommodate safe and street food, and our team and we will use this information and are going to conduct a survey. Yeah, thank you for your patience listening. Thank you for the opportunity.
Yeah, thank you. My question is, what is the relevant aspect in a street food? Most relevant aspects.
Ma'am, most relevant aspect is a health, ma'am, because street foods are consumed on a wide range, and that poses a lot of health risk. So
So, so
so ma'am, like this is the aspect we are focusing on. Yes, ma'am.
do they require any kind of trainings, or do they require any kind of awareness?
Yes, ma'am. Yes, ma'am. We need a lot of training and awareness, and that is why we need like many initiatives around. I mean, FSI is working on clean street food, and we have many other initiatives. But on a personal level, also vendors are supposed to get the digital training provided by FSI. That would really help. And also, consumers should avoid consuming unsafe street food. There are many street foods which are also like safe. Vendors are encouraged um, to follow all the protocols and all. So yes, ma'am, there should be a lot of awareness and many more initiatives to make the street food safe. Safe. Yes, ma'am.
So food hygiene and hygiene aspects are the important for
Yes,
this
ma'am.
business. Okay, thank you
And
so
also
much.
the raw materials and the preparation methods, ma'am, because oil or other materials are like reused, and that also poses health effects. Like
So that is quality, no? Quality of
yes, ma'am,
ingredients,
quality.
quality of raw material. Thank you.
But have you conducted any survey on food safety,
Ma'am, could
or
you
is
please,
it a theoretical aspect?
ma'am, could
Have
you
you conducted any survey to check
ma'am,
the food safety?
ma'am, we are about to conduct a survey.
Have
Like
you
since
surveyed the food?
ma'am, we are about to
Have
conduct
you
a survey,
surveyed the
ma'am.
food business operators?
Ma'am,
So,
could you,
your study is based on what parameter?
ma'am, our studies are based
Your
on the
study is based on what parameter?
ma'am, our studies is based on the previous researches, and we've spoke to like few street vendors. We didn't do like proper research, and then we have considered other studies, news, and also the different perceptions of the consumers in our class in a very small scale, which is not very legit survey. But we are going to conduct a legit survey very soon, ma'am. Since the classes are offline now, we'll do that like very soon.
But okay, fine then. Next.
Thank you, ma'am.
Thank you. Now I would like to welcome the next group of presenters, Thota Akansha and Bushra Shehda, on the title of abstract, Influence of Nutrition Education, Intervention of Nutritional Status of Cancer Patients.
Good morning, ma'am. Let me share my screen. Am I audible?
Yes.
Yeah. Good morning, everyone. And my title of the presentation is A Study of Nutritional Status of Cancer Patients in Shada Hospital and Educating Them. And I'm Akanksha, and my supervisor is Bushra Shahida, Assistant Professor of Shada University, Greater Noida. This presentation mainly contains introduction and why is this research essential and how is the research conducted and the data and the conclusion of the research. First of all, what is cancer? We can say that cancer is a disease condition by which an uncontrolled division of abnormal cells take place in a part of the body. And we also know that the cancer is one of the leading cause of the mortality. And the few risks of cancer are diet, alcohol, physical activity, hormonal factors, radiations, etc. And it is estimated by previous studies and research that 30 to 40% of the cancers can be prevented by appropriate diets, physical activity, and by maintaining appropriate body weight. It was also known that the food components influence cancer both positively and negatively. Methodology. actually in this for conducting the research, we have followed two phases. In the phase one, we studied the socio-demography and disease profile and nutritional status of the cancer patients who are visiting Shada Hospital. And also in the phase two, we educated the patients about the nutritional importance in the cancer by using a brochure. In the phase one, we prepared the questionnaire and collected the data. In the questionnaire, we included mainly the socio-economic data, demographic data, anthropometric data, biochemical parameters, lifestyle of patient, and clinical signs and symptoms of the patient, and nutritional assessment of the patients was done by using MNA, that is mini nutrition assessment form. By this, we collected the data, and for the collection of data, we selected a group of patients who were visiting the Sharda hospital for their treatment, and we had done their interview both on call as well as and face-to-face interview. In the phase two,
Akanksha,
yes, ma'am.
sorry for interruption. You have only 1 minute to conclude.
Okay, ma'am. Okay, and we educated in the phase two, and in the phase two, for the education purpose, we use this brochure that we have made ourselves. Sorry, in which we mainly included the food groups and dietary components, which help in competing the cancer and helping for the treatment of the patients in the cancer treatment. And this is the data that we collected from the patients. Those are visiting the Shada hospital, and different types of cancers they are prone to, and different stages data that we collected. And in this we can see that the cancer patients are categorized into 3 stages, that is primary stage, that is stage 1, stage 2, and stage 3. In the stage 1, we have assessed the nutritional status of the patients and categorized based on their nutritional status. There is normal, risk for proning malnourishment, and malnourished. In all the stages, we have made this chart so that we can compare the data of the nutritional status of the patients in different stages. And from this,
Akanksha, please conclude your
yes, ma'am. In this, we have studied that as the patients visiting the Shada hospital are not in a wide range, thus we were not able to collect more data. But as for the data that we have collected, we have concluded that the patients suffering from stage 1 cancer are mainly at the risk of attaining the malnourishment, whereas in the stage 2, not many people were in the stage 2 that were visiting the Shada hospital. But as per the similar research that we referred, we can say that the malnourishment nutritional status of the patient increased in the stage 2, and in stage 3 we concluded that the level of subjects malnourished increased compared to the stage 1. And this is due to the less awareness of the knowledge in the subjects in relation to the nutritional importance in the cancer, due to which the deteriorating of the nutritional status of the patients have been concluded. Thank you.
Thank you, Akanksha. Any questions?
Yeah, Akanksha, my question is, what were your determinants for this study?
Ma'am, basically, for the assessment of the nutritional status, we used a form that is mini nutritional assessment form, in which it basically contains the data of the weight, like before in different stages of the cancer, like what was the weight and did they lose any weight, and their appetite and their nutrition habits, like they're including any sort of antioxidants or fiber or pulses or proteins, or some questions that help us understand the nutrition intake of the patients.
Okay, so how you have done sampling, and how many patients you have studied?
Ma'am, right now we have studied about 25 patients, and we are further going to study more people. But right now we have a sample size of 25, and this study is basically conducted mainly on face-to-face basis. Like the interview has been conducted by visiting the patient.
Okay, thank you.
Thank you, ma'am.
Okay, thank you, Akanksha. Next participant, please.
Thank you. Now I would like to welcome the next presenter, M. Agarwal, for oral presentation of title of abstract, Infant Feeding Practices in a Slum in Delhi.
M. Agarwal, are you present? Okay, we will move with next student.
So the next presenter is Ms. Rachna Dutt and Dr. Payal Mahajan for their oral presentation of their title of abstract, Impact of Education on Colostrum Feeding Practices Among Nursing Mothers.
Okay, Ms. Rachna, are you present? Okay, go with next student.
So the next presenter
Go with next student.
So the next presenter is Ashmita Singh for their oral presentation of their title of abstract, Cordia dictoma, G. Forst, an underutilized traditional plant.
Okay, Ms. Ashmita Singh. Okay, I think she's also not present in this session. Then we will move to next participant.
Now I would request Ivan Matthew to carry forward the session.
Okay, meanwhile, Ivan is joining. I'll call next participant, Shalvika. Shalvika is there?
Yes, ma'am, I am there.
Okay, Shalvika.
Ma'am, may I share my screen?
Yes, sure.
Ma'am, is my screen is visible?
Yes, it is visible.
Okay, ma'am. Ma'am, I am a PhD scholar from Sharda University, and my topic is starch modifications, methods, and its application. It is just a review article, and in this, starch is a polysaccharide found in plants. They are also known as amylum. It constitutes a major food supply to humans all over the world. It is an essential food product as well as a biomaterial that is utilized for a variety of uses all over the world. The important sources of starch for the living creatures are root, tubers, seeds, rhizomes, grains, and cereals. Starch is also the primary sugar stock in all the cereals, rhizomes, tubers, as well as organic products, which comprises of straight chains of amylose, that is 25 to 35% in proportion, and exceptionally stretched amylose pectin chain near about 65 to 75%. Starch does not have physical and chemical properties suitable for certain types or processing in its native form due to their poor shear and thermal stability and high degree of retrogradation. Several properties may be altered by modification, including the gelatinization properties, swelling properties, solubility, adhesion, and other properties of the native starches. The new ingredients can be used in many processes and products with more desirable properties than in their original form. Starch granules. In view of the great diversity in starch granule morphology, it is remarkable to find that their internal architectural features are shared universally among the plants, regardless the plant organs. When it is observed in the cross-polarized light, it is in optical microscope, a maltase cross is typically seen extending the arms from the so-called hilum, which is believed to be the origin of growth of the granules. There are two parts from which starch is made, that is amylose and amylopectin. Amylose are the linear chains with 500 to 2000 glucose units, which constitute of 20% of the starch, and it is soluble in water. It contains alpha 1,4 glycosidic bonds between two glucose units. Amylopectin is a highly branched chain within over 1 lakh glucose units, constitutes of 80% of starch. It is insoluble in water, and it contains both alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bond at the branchings. Why starches are modified? Starches are modified to enhance their performance in different applications. Starches may be modified to increase
Shalvika, sorry
their
to
stability.
interrupt. You have only 1 minute to compile your presentation.
Okay. The main method of starch modifications are enzymatic methods, chemical methods, and physical methods. If we go through the physical methods, there are some physical methods that are heat moisture treatment, annealing treatment, UHP, microwave, ultrasonics, and so on. For chemical modifications, we have three methods: oxidation, esterification, etherification. Enzymatic method, in which we use one or more enzymes to modify the starch. These are the schematic representations of enzymatic modifications. Applications. What are the applications of modified starch? It is used as a self-healing polymeric material. It is used in the process of delivery of drugs as a starch-based films in food packaging. It is used in canned foods, baked foods, and frozen foods also. This flowchart represents how modified starch markets. What are the functions, what are the applications, and what are the raw materials? The major findings are the branched major component amylopectin is generally agreed to be principal contributor to the semi-crystalline structure of the granules. Polymers with viscoelasticity and extremely elastic module stages could be equated with some additives within the polymer to protect the starch from unbinding issues. This could retain, avoid, and prevent further growth of these breaks that supplied in the small and widely dispersed areas. What are the conclusions? Starch is rarely consumed in its native form due to the restricted solubility in water, heat resistance, gelatinization, and etc., which limits the industrial applications. With the world focused on moving towards sustainability, starch has found itself as one of the forefront runners in the development of functional materials for advanced applications. The future prospects. There is a current trend by improvement of techniques and processes using innovative green methods for the starch modification. Besides promoting changes in the physical, morphological, and rheological properties, bringing new functionalities that are not found in the native starches. These methods have the advantages of not using chemical reagents. Bioplastics are also currently experiencing strong growth worldwide due to the
Shalvika, you have already exceeded your time limit.
Okay, thank you, ma'am.
So, any questions? Okay, so we'll move the next participant.
Now I would like to call S.A. Marak, N. Kaushik, A. Dickey, E. Suminira, and E. Faj. And the topic is optimization and development of muffin from hibiscus subrefa calyx extraction using RSM.
Can I start? Ma'am, am I audible?
Yes, you are.
Yes, you are audible.
Okay. Good afternoon, everyone. Myself, Sengnolothai Emarak. I'm pursuing my PhD in Foods and Nutrition from MIT University. And my topic of presentation is optimization and development of muffin from hibiscus subdarifa calyx extract using RSM. Hibiscus subdarifa, commonly known as roselle, is an annual herb that grows up to 7 to 8 feet They are mostly cultivated for leaves, seeds, flowers, and calyx. The flower of roselle is similar to that of cranberry with sour and tangy taste. Its uses and applications are, it is widely used in foods and beverage industry, pharmaceuticals, cosmetics, and in folklore medicines. The health benefits of roselle are well-known nutritional and medicinal properties due to its phytochemicals and bioactive compounds. Many researchers have pointed out that roselle calyx extract possess many health benefits, such as blood pressure lowering effect, lowers cholesterol level, anti-diabetic properties, antioxidant activity, and also it contains several amino acids that are important for our body. The rationale of the study is nowadays addition of natural extract from medicinal plants to food products are becoming very popular due to its abundant content of bioactive compounds. Roselle is one such plant whose aqueous extract contains rich red pigment due to the presence of anthocyanins. Well, color in any food, it plays a major role in consumer acceptance of any food. Roselle calyx appears to be good and promising source of water-soluble red colorant that could be utilized as a natural food colorant. However, in any attempt to formulate unusual ingredients in food product, it needs to be first characterized by a comprehensive approach using statistical tools in order to determine the composition. Therefore, for this reason, RSM was used. The study design or the experimental was designed using box Pencan RSM design using design expert. This generally consists of preparing the response design, formulation, response analysis, and optimization. In this study, three independent factors or component, that is extract volume, citric acid, and sodium bicarbonate was used for responses, texture, and overall acceptability score. The formulation of roselle muffin was carried out as shown in the flow diagram. The raw material used was dry roselle calyx powder. Then the extract was prepared using aqueous solution. After that, the batter was prepared in the proportions as obtained in the experimental design to form different formulations and baked at 170 degrees Celsius for 30 minutes. Then the next step is response analysis, which was evaluated by sensory preferences using 9-point hedonic scale. The range of score was 6.36 to 7.23 for texture.
Sorry to interrupt, Mark. You have only 1 minute to conclude your presentation.
Okay, ma'am. This is the sensory evaluation. Then the next step is optimization, where it was carried out. And here the optimization goal was to maximize the extract volume and texture. Here we see the optimization indicating that the following values with a desirability of 0.840, which turned out to be higher as seen than the experimental value. Then the nutritive value of the optimized muffin was also determined. We see that optimized muffin is low in fat, protein, and total carbohydrate as compared to control muffin. However, the ash and vitamin C content was significantly higher than the control muffins. Then the antioxidant activity of roselle muffins and extract was also determined to check the stability of the phytochemicals after baking, because we all know that temperature is one major factor that affects the antioxidant activity. Here we see the heating caused an acceleration of the initiation reaction and cause a decrease in activity of the present phytochemicals. However, fairly good amount of phytochemicals are still retained in roselle muffin. To conclude, I would like to say that the optimization was successful for development of roselle muffin, resulting in high nutrition, good amount of antioxidant, ascorbic acid, that has the potential of having many health benefits. And also the results can be a valuable information for developing food products using roselle calyx extract. Thank you.
Any questions?
Next, ma'am, because we have short for time. So.
Okay, ma'am. So I would like to invite Shakshi Verma, Roji Behra, Rhythm Bhardwaj, Rashmi Yadav, and Prabjot Sabrawal. And the topic is biodegradable 3D pulp containers trays from banana peel waste.
Sakshi, are you present?
Good morning, everyone. Am I audible?
Yes, you are audible.
I just share my screen first. Just a second. Okay, am I visible to you all?
Yes.
Good morning, everyone present here. I am Sakshi Verma from Shahid Rajguru College of Applied Sciences for Women. Today, on behalf of my team, I am going to present our research work on the topic biodegradable 3D pulp trays and containers from banana peel waste. Let's start with the introduction part. Fruits and vegetables are the most produced and utilized commodities out of all the horticulture crops. India is the leading producer of fruits in the world. Significant losses and waste in the fresh and processing industries are becoming a critical nutritional, economical problem as well as nuisance to the environment as solid waste. According to the United Nations Food and Agriculture Organization, the losses and waste in fruits and vegetable production are the maximum among all types of food and are as high as 60%. Around 12 million tons of fruits and 21 million tons of vegetables are wasted each year. Despite their pollution and hazardous effects, food processing waste have a great potential for conversion into a value-added product. Now, what are the objectives of our research? The objective of our present work was to study first the effective management and utilization of banana peel waste, production of a 3D pulp trays or containers from banana peel waste, and then last, characterization of the packaging material. Now, the methodology we use. We procured our banana peel from the local vendor of Mayur Vihar Phase 3, Delhi 96. Then the collected peel paste was grinded to form paste, heating of the paste to inhibit microbial growth. With the help of a muslin cloth, we get the peel pulp. The collected peel pulp was then molded in a 3D egg tray and different containers. Then 3D trays and containers were dried at 85 degrees Celsius for 2 to 3 hours. Now, what are the results we get? Biodegradable egg trays made out of banana peel is formed nicely as an alternative as in the commercial market. The appearance of the containers and egg trays prepared by the banana peel was found out to be dark brown or black in color and were molded well. And it was found that peel was perfectly get a shape after drying as compared to the original molds. Black color of the banana peel extract is due to the change in the temperature of the environment and the chemical reactions occurring on the skin of the banana. Also, this is cause of the enzymes too. We also tried to develop some containers of different shapes out of banana peel waste. The containers and the trays were tested for moisture content, GSM, and thickness. 3 trials were done for each of these parameters, and the values obtained were compared with those of the commercial pulp-based trays. Biodegradation studies were conducted on the prepared containers and trays, which showed a decrease in the weight of the mold kept in the soil day after day. The developed biodegradable containers are much cheaper than the paper pulp trays. In case of paper pulp trays, the cost of paper also must be considered. However, no extra investment is needed here as we have used the banana peel waste as our raw material. In costing, only labor charges and drying charges in case if we are using oven drying method are included. Now, the thickness, the GSM and the moisture content we calculated were same as that of the commercial tray, and it's preferable for the commercial level production. Now, these are the pics of our egg trays. These are the biodegradable 3D egg trays from banana peel waste, and these are some of the containers from banana peel waste. Now the conclusion part. Issues concerning environmental impacts of food packaging waste have prompted development of biodegradable materials, generally from renewable sources. In this study, egg trays and containers were prepared by using banana peel waste and were characterized by using several testing methods. Based on all performed standard methods, visual inspection, and degradation studies, it can be confirmed that commercial egg trays and containers for packaging can be replaced by those developed from banana peel waste. Thank you so much. I hope it is totally understandable to all people.
I have one question for you. Have you studied it microbiological aspects?
Yes, ma'am. Sorry.
Have you studied this trace on the aspects of microbiological?
No, ma'am. Further, we didn't did yet, but we are looking forward to do this. The biodegradable.
What is the shelf life of this tray?
Ma'am, the shelf life actually it is not like it is not get affected. So the shelf life may be longer, just like the pulp trays.
So you have not studied that, right? You have not studied that.
No, ma'am. Actually, the shelf life is like we kept it for 3 weeks, and it is almost similar to the first day, so that we assume that the shelf life is not affected.
Okay, thank you. Next, please.
Now I like to invite Nisha, Kautham, Agrawal, and Singh. And the topic is development of carambola candy by osmotic dehydration.
Nisha, are you present? Nisha? Okay, Ivan, we will proceed with the next student.
Okay, ma'am. Now I like to invite Mudassir Ahmed, and the topic is starch-based nanomaterials and the delivery system for encapsulation of bioactive compounds.
Thank you very much for providing me with this opportunity. I'm Dr. Mudassir Ahmed from University of Kashmir. Is my this slide visible to all of you? Hello?
Yes, it is visible.
Okay, I'll be presenting my work related to starch-based nanomaterials and delivery systems for encapsulation of bioactive compounds.
Yes, Mudassir, please go ahead.
Srinivas, meanwhile, should we go to the next participant?
Ivan, I think he has some connectivity issues. So we will move to our next student.
Okay, ma'am. Now I'd like to invite Gargi Teku, Bhagat Singh, Charu Gupta, and Girish Sharma. And the topic is antioxidant bioconstituents from agro-industrial waste and their importance in functional foods.
Yes, ma'am, I'm sharing my screen, just a minute. Is my screen visible? Hello.
Yes, ma'am.
So good afternoon, everyone. I'm Gargi Teku, and I'm a student of BSc Honors Medical Biotechnology from MIT Institute of Biotechnology, MIT University, Noida. and I'm currently working on the agro-industrial waste, and I'm here to present my work on the antioxidant bioconstituents from agro-industrial waste and their importance in functional foods. So our work is aimed to find a way to treat or overcome the oxidative stress, which is primarily caused by the ROS. ROS, or the reactive oxygen species, are free radical species, and they have very high tendency to react with the available biomolecules and affect their function and structures. They majorly affect the structures of DNA, RNA, and proteins. They are mainly caused by either extrinsic or by the intrinsic factors. The extrinsic factors include xenobiotics, microbes, radiation, growth factors or cytokines and chemokines, while the intrinsic factors include metabolizing enzymes, endoplasmic stress, or mitochondrial pathways. These ROS majorly cause oxidative stress, which in turn causes a number of disorders like inflammation, aging, and cancer, etc. To overcome this stress, our body has a natural oxidative stress defense mechanism. This includes DNA repair machinery and physical defense barriers like the skin and the cell membranes. The body also tried to overcome the production of these reactive oxygen species by chelating metals. Another machinery working against the oxidative stress is the antioxidant defense, which includes enzymatic and non-enzymatic antioxidants. Our study focuses majorly on the non-enzymatic antioxidants. These can be further divided into various subtypes, and these are dietary sources including vitamin A, vitamin E, vitamin C, and carotenes. Direct and indirect acting low molecular weight antioxidants, and the main focus of this work, the phytochemical including the polyphenols and bioflavonoids. The samples which were used in this study were the allium cepa, which are the onions, and the Arachis hypogea, which are the peanuts. We studied the presence of these polyphenols in terms of gallic acid equivalents. To achieve this, we collected the sample from nearby available sources and prepared their extract. This extract was made in 50% methanol, and further we estimated the total phenolic content of the collected sample by the following method. The absorption of the samples are taken up by the spectrophotometric method. These are the instruments that were used in this study. The analysis of the chemical composition and their activity was analyzed by the spectrophotometric method. Further moving on to the results, the total phenolic content in the seed coat of peanuts were 64.1 mg per gram, whereas in the peels of onion were 57.5 mg per gram. The total phenolic content was shown high in the seed coat of peanut, and the amount of antioxidant activity in residues was from 27.8% to 89.8%. The agro-industry processing waste of fruit and vegetable are good sources of bioactive phytochemicals, and value-added substances like polyphenols are important for their antioxidant activity. Such waste have high value as food or feed after proper treatment. Thus, such agro-industrial waste can be cost-effective, eco-friendly source of bioactive antioxidant phytochemicals, and the antioxidant provide protection from damage caused by the reactive oxygen species that can be used as supplementary functional foods, which can be beneficial in treating and in preventing various lifestyle diseases. Thank you.
Any questions?
Next, please, ma'am. Next, please. Next.
I'd like to call back Dr. Murasir Ahmed for his presentation.
Sorry for the inconvenience. Previously, there was interruption with my internet connectivity. I'm Dr. Mudassir from Department of Food Science and Technology, University of Kashmir. I'll be presenting my work related to starch-based nanomaterials and delivery systems for encapsulation of bacterial compounds. This is my educational background. I will straight come to my research. Starch, you might be knowing that's a biopolymer, and it has tremendous food applications. And it comes from the several sources, but in native form, it has several limitations, like it has solubility, it faces retrogradation, it has limited digestibility and poor functional properties. Starch has been provided with several modifications like physical, chemical, or enzymatic modifications. And with these modifications, there's improvement in the starch, and applications have been improved in several this food or pharmaceutical or textile industries. But there's a novel technological approach by which we can modify the starch, and its applications are now been increasing with a greater pace. It's a nanotechnological approach by reducing the size of the starch macromolecule to the nano level. Our focus of study is starch nanoparticles. We have selected the starch from horse chestnut, water chestnut, and lotus stem. What are the expected applications? We are expecting there's improvement in techno-functional and nutritional properties of the starch. We expected to utilize it as a nano delivery vehicle for encapsulation of active compounds, so that we can protect those bioactive compounds against the digestion conditions. Starch nanoparticles have been modified by using, our approach was to use the green technology or the mild chemical treatment. Then we utilized it for encapsulation of bioactive compounds. The bioactive compounds, they have the low viability, they have less permeability, and they often degrade under digestive conditions. What the encapsulation will do, it protects the functional components, these bioactive compounds, against the gastric conditions, and improves their viability and permeability. In one of our study, I will just highlight the outcome of my studies. In one of our studies, we prepare the starch nanoparticle by ball milling. And what we observe is that the starch nanoparticle, it had a high water absorption and less oil absorption capacity than native starch. It means that we can use the starch in a very less amount, or it can absorb a very less amount of the oil that will be beneficial from the health perspective. And further, these starch nanoparticles, they were amorphous, there was increased viscosity and better thermal stability. Increased viscosity means that we may require a less amount of the starch to achieve a desired viscosity in several food products. In another study, we developed the starch nanoparticles by mild alkali hydrolysis and ultrasonication processes. This slide shows the preparation of the starch nanoparticles. I will just come up with the results. And the starch nanoparticles, it was successfully synthesized by using the mild treatment combinations of alkali hydrolysis and ultrasonications. Starch had better thermal stability, increased viscosity, and good antioxidant properties. It were more amorphous, decreased oil absorption capacity, and increased water absorption capacity. There was a decrease in the crystalline nature of the starch. It was more amorphous. And then comes to this encapsulation. We utilize these starch nanoparticles as a tool for delivery of active compounds. In encapsulations, I will skip this slide so that we can save the time. We use it, there are several techniques for encapsulation, like drying, freeze drying, supercritical fluids, and these are the techniques we are most interested in. In one of the study that we encapsulated catechin in starch nanoparticles. And why to encapsulate catechin? It has a potent antioxidant property better than butylated hydroxytoluene or butylated hydroxytoluene. It inhibits several carcinogenesis processes. But the major shortcoming for this catechin is its low viability, less permeability, and it degrades under digestive conditions. It undergoes the auto-oxidation reactions in the lower part of the digestion conditions. It has several sources like berries, apples, grapes, pears, but green tea is considered as
Dr.
one of the
Muddassir, sorry to interrupt, but you have only 1 minute now to conclude your presentation.
Okay, sure. I will be just presenting the outcome of my studies. This is the process how I prepare the starch nanoparticles, and we studied encapsulation efficiency, zeta potential, structural characters by several techniques, and then bioactivity retention via antidiabetic and anti-obesity properties. What we conclude that nano capsules of catechin, they are more beneficial than free catechins. There was slow and controlled release of catechin in gastrointestinal conditions. The bioactivity was better retained after digestional conditions. In another study, we encapsulated resveratrol. Resveratrol is a highly anti-carcinogenic, anti-inflammatory, anti-obesity, and antidiabetic properties, but it's unstable to light, heat, or enzymes. It is more active in trans form, and upon the exposure of the light, it converts into the less active form, into that is cis form. We studied its structural characterization by SEM, FTIR, and XRP. The starch, these nanocapsules of the resveratrol, they were more amorphous, and FTR and SEM shows the presence of the resveratrol in the cavities. The encapsulation efficiency was really around 60%, and the total amount of resveratrol retained was, you can have a look at the data. I will move to the next slide. And what were the outcome of the studies? That you can see the pancreatic lipase and cholesterol esterase inhibitions. It was much retained under encapsulated conditions and free from. It just shows the 26 to 23%, but upon encapsulation, it was improved to a higher level. What are the applications in the food industry? We utilize these resveratrol nanocapsules for development of functional snacks. These functional snacks, they had the antidiabetic, anti-obesity, and antioxidant. But we conclude that
we will not be able to give more time, as there are the other, we are already running behind schedule. So it's a request that you have to please, within 1 or 2 lines, please summarize what the
Sure, I will be concluding with this slide that these resveratrol capsules in snacks, it shows the better antidiabetic, antioxidant properties than the snacks which contains the free resveratrol. The sensory analysis was equivalent. There was not much difference in the sensory of the snacks. The antioxidant and antidiabetic properties were higher in snacks contain encapsulated resveratrol. Encapsulation process can prevent the thermal degradation of bacteria during processing. The study highlights the nutraceutical importance of using bioactive compounds in an encapsulated form in various food formulations. This is the future scope of these studies. We can go for the bioavailability and cell permeability tests. We can utilize these bioactive compounds in different food formulations. We can check the viability under in vitro or after consumption of the foods when we incorporate or fortify the foods with these. And
very nice presentation. Yeah, thank you.
Now I like to invite Hardik Gupta, Bhagat Singh, Charu Gupta, and Girish Sharma. And the topic is antioxidant phytoconstituents from agri-horticulture waste and their role to combat oxidative stress and associated disorders.
Hello. Am I audible?
Yes, Arden.
Yes, ma'am, I'm presenting. Is it visible?
Yes, it is visible.
Good afternoon, everyone. I'm Hardik Gupta from Medical Biotechnology, BSc Medical Biotechnology, and I'm studying in MIT Institute of Biotechnology. And my topic for today is antioxidant phytoconstituents from agri-horticultural waste and their role to combat the oxidative stress and associated disorders. So the main target of our study was to overcome the oxidative stress, and to understand oxidative stress it is basically the imbalance between the production of ROS and the antioxidant defense mechanism of our body system. So to understand ROS, they are basically the free radicals, and they could be induced by various metabolism, heavy metal, UV light, pollution, smoking, radiation, etc. And these are mainly inhibited by antioxidants. While when we are talking about the effect of oxidative stress, then this may produce lipid peroxidation, protein damage, or DNA damage to the cell. And if we see the mechanism how it is happening, so when normal cells is under ROS, like when there is more production of ROS in the normal cell, so the free radicals attack the cell, and this causes the oxidative stress in it, which further causes apoptosis or necrosis in the cell. And these are mediated by the protein damage and the DNA damage. So when we are talking about the associated diseases, so free radical oxidative stress causes diseases to almost all the body parts, and there are many multi-organ diseases like diabetes, aging. There are diseases which could be like cardiac fibrosis and chronic kidney diseases. They could also be like it has been seen that oxidative stress has significant role in cancer, like manifestation of cancer. Now, further talking about the antioxidant defense, so there could be two types of antioxidant defense. It could be enzymatic or non-enzymatic. In non-enzymatic, it could be of low molecular weight antioxidants, or it could be plant antioxidant. So as I'm particularly working upon the plant antioxidant, so it could further be of two types, which could be dietary sources, and these are various vitamins like vitamin A, E, and C, and there could be carotenes also. And when we are talking about the phytochemicals, so they are basically the flavonoids, bioflavonoids, polyphenols, and other ones. So I basically mainly studied upon like calculating the polyphenols, and these were the major samples which I took, which were orange, marigold, pot marigold, papaya, and rose. And I prepared an extract in the 50% methanol, and I performed TPC, which is total phenol content, and antioxidant activity in these plants. And for the total phenolic content, we use Follin's method, and for antioxidant activity, we use BPPH method. So as my study is like still in very preliminary stages, and it is not yet published, so I'm not sharing the whole of the result, but like the antioxidant activity which I got was ranging between 57 to 95%. And while the TPC which was there, it was like approximately from 105 milligram per gram equivalence of gallic acid in the like which was the lowest ones, and in the highest which I got till now was 256 milligram per gram. Talking about the conclusion of my studies, so we have seen that there is significant presence of phenolic content in these agri-horticultural waste, and as these wastes are generally just disposed of and they causes the pollution, so if we utilize these waste in some nutraceutical methods or maybe as functional food, so they could like they could help us in prevent these problems of disposal and even pollution also. And the antioxidant activity has shown that the like these can be used as functional foods and as nutraceuticals. These are some of the references which are used. Thank you very much.
Thank you. Next, please.
Now I would like to invite Ms. Akansha Agrawal to continue with this session.
Thank you so much, Ivan. Moving ahead, I would like to invite Asha Kumari, Nutan Kaushik, Raza, and Khushbu to share their presentation on the topic production of protein hydrolysates from surmi processing byproducts using enzymatic hydrolysis.
Good afternoon, everyone. Myself, Asha Kumari. I am pursuing my PhD under the guidance of Dr. Nutan Kaushik. Today, I am going to present on the topic production of protein hydrolysate from surmi processing byproduct using enzymatic hydrolysis. And during surmi production, 60 to 75% of fish byproduct are generated. These byproduct are either dumped back to the sea, which cause serious environmental pollution and harmful effect on the public health due to the release of organic and inorganic compound, or it is used for the production of low value products such as fish feed or silage. This byproduct are the resource of protein, collagen, gelatin, and omega-3 fatty acid. Therefore, the feasible technology are required for the proper utilization of fish industry byproduct in the economical and ecologically sustainable way. This not only help in eliminating the harmful effect on environment, but industry can earn extra profit by producing value-added product with the help of feasible technology. Technology such as enzymatic hydrolysis has the potential to produce high-value product from the fish byproduct, which can be used to fight against the malnutrition and nutrition-related disease. In this study, pink perch byproduct was procured from surmi processing industry. Then the chemical composition of byproduct was determined, and it was observed that the head and viscera contain 76% moisture, 2% lipid, 5% as, and 70% protein, and skin and bone contain 69% moisture, 2% lipid, and 7% as, and 22% protein. From the chemical composition analysis, it was observed that the byproducts are the resource of protein which can be used for the development of different value-added products. Before going for the enzymatic hydrolysis, the byproduct was first analyzed for the presence of endogenous enzyme, and it was observed that the head and viscera contain high amount of endogenous enzyme as compared to the skin and bone. Enzymatic hydrolysis of pink perch byproduct was performed by using different enzymes such as alkylase, trypsin, bromelain plus papain, and endogenous enzyme. The condition for the enzymatic hydrolysis are at 50 degrees centigrade for 1 hour, and enzyme to substrate ratio is around 0.1% of the raw material. After performing enzymatic hydrolysis, protein hydrolysate fraction was characterized for yield percentage, degree of hydrolysis, antioxidant activity, and amino acid composition. From yield percentage, it was observed that the high yield of protein hydrolysate was observed after alkylase hydrolysis as compared to other enzyme. Degree of hydrolysis of protein hydrolysate was determined by formaldehyde titration method, and it was observed that in case of head and viscera, there is no significant difference between the degree of hydrolysis of protein hydrolysate obtained after hydrolysis with commercial enzyme, but it was significantly different from endogenous enzyme. But in case of skin and bone hydrolysate, there is no significant difference between the protein hydrolysate of alkylase and bromelain plus papain, but was significantly different from trypsin and endogenous enzyme. Antioxidant activity of protein hydrolysate was determined by DPPH scavenging activity, and it was observed that all the protein hydrolysate was showing moderate antioxidant activity. Amino acid analysis of protein hydrolysate was determined by HPLC method, and it was found that the protein hydrolysate obtained after alkylase hydrolysis has higher amount of essential amino acid composition as compared to other enzymes. Protein efficiency ratio of protein hydrolysate yes, ma'am.
You have only 1 minute to complete.
Okay, ma'am. Okay, ma'am. Protein efficiency ratio of protein hydrolysate showed that the high quality protein was obtained after enzymatic hydrolysis of all the commercial enzymes. At last, I can conclude that the enzymatic hydrolysis is a potential and sustainable technique for the utilization of surmi processing byproduct. Pink purge byproduct are the potential source of high quality protein as a protein hydrolysate, which contain high amount of essential amino acid, which is further used for the development of different value added product. Thank you.
Next, please.
Next, I call Dr. Vandana Singh to share her presentation on the topic Evaluation and Green Synthesis of Bioethanol from Valorization of Food Waste.
Dr. Vandana, are you there? Okay, so Akansha, we will proceed with next candidate.
Okay, ma'am. Moving ahead, I call Ranjana Surolia and Anuradha Singh to present their presentation on the topic Valorization of Food Waste, Sources and Applications.
Okay, good afternoon, everyone. I'm going to share the screen. Am I audible? Am I audible to everyone? Is my voice is audible to everyone?
Yes, you are audible.
Thank you so much. Okay, good. A very good afternoon to everyone. Myself, Rajini Sirodia, a PhD scholar at Galgotia University under the supervision of Dr. Anuradha Singh, professor at Galgotia University. And my topic of presentation is valorization of food waste sources and applications. This is just a small review just to convince everyone to how to maximize the research on utilizing the food waste in different applications. So let's begin with the introduction that every year 1/3 of global food are discarded or lost, which led to the major environmental issue. And that is really a problem which need a concern. Maximum discarded food waste ends up in a landfill, which result in the production of greenhouse gases such as methane and carbon dioxide, and which causes the global warming. So appropriate quantified methods are required to reduce the amount of food waste, but that is not enough because this is an unavoidable phenomenon. So alternative use of food waste is a need and demand of the present environmental condition. In recent years, there is a great emphasis on efficiently reutilization of food waste, or we can say agro waste or solid industrial waste. But why there is a need of reutilization? Like there's only environmental issue, that is the reason? No. Food waste are a rich source of natural bioactive components such as antioxidant, antimicrobials, vitamins, and many other nutritional properties. And it can be exploited as a source of high-value components such as protein, fibers, polysaccharides, flavor compounds, or different phytochemicals. So they can be valorized as functional ingredient in food, pharmaceutical, healthcare, cosmetic, and other products also. So food waste exploitation is an inexpensive and sustainable alternative for isolation of many bioactive compounds or molecules, and they are useful in production of health promoting products, and also at the same time contributing to an efficient way to minimize the environmental issue and also offer the economical growth. If we talk about some sources of food waste, starting from harvesting till the consumption, there is a small amount of food waste which occurs at every step. Like in industry, if we talk about the food processing industry, they generate approximately 45% of total industrial organic pollution. And in the food supply chain, starting from field of production, processing, distribution, consumption, and disposal, there is some extent of waste which is taking place at each and every step. There's basically two types of wastage which generated from the food industry, that is animal derived and plant derived. Animal derived categorized into further three, that is meat product, dairy product, and fish and seafood.
Plant-derived like cereal, for example, rice bran, wheat bran, etc. Fruits and vegetable, orange peel, grape pomace, apple pomace, etc. Root and tubers, that is potato peels, sugar beet, etc. Oil and crops, oil crops and pulses, that is sunflower seeds, soya bean.
You have
Yes,
only 1 minute to complete.
So I'm not going to discuss this step, that is starting from total harvest to consumption. There is a small amount of waste which is occurring in each and every step. I'll come to the application. Some application is already discussed by my co-participants. So food waste are rich in biodegradable material, making them suitable substrate for number of applications. It can transform into energy, and this energy can be used in a hydroelectrical energy. Chemical modification is also possible. There is a range of products such as biofertilizer, bioplastic, biofuels, chemical and nutraceuticals which can be come from this food waste. They can be used in biotechnology also, such as hydrolyzed and glucose, free amino nitrogen phosphate, which can be utilized by many microbes. And chemical modification of this organic waste also reduces the problem of resource depletion. Nutrients such as sugar monomers, etc., are used for the cultivation of bacteria, fungi, algae, and the production of many metabolites. Green chemical become green chemical technology.
You have
And this is just the
already
last slide. I'm just concluding this. This is
Okay.
just the last slide, ma'am. This is just a small review, just to aware everyone. So, valorization of food waste from green chemical technology that diversify the production of many useful products. I'm not going to categorize every, but I'm giving a small example that is citrus waste can be converted into lemonade, that is a fragrance or a solvent or chemical intermediate. Terpeneol, that is basic ingredient used in a perfume. Pectin is very useful polysaccharide which is used as a thickening agent, stabilizer, prebiotic, edible coating in the food industries. And there are many other byproducts which can be used in different industries. I conclude this that the food waste reutilization is a cost-effective, sustainable alternative and also offer the economical growth. So requirement, what is the requirement of present scenario? That the contribution from various disciplines like engineering, biotechnology, environmental science, economics for complete utilization of it. And this waste product, chemicals, and food industries also. These are the sub-references. Thank you.
Thank you, Rani.
Thank you. Next, please. Next, please.
Moving ahead, I call Asli Kan Karaka. I apologize if my pronunciation was inappropriate to present, and the title of their presentation is Protein Extraction from Waste and Byproducts of Food Processing.
Good morning. My name is Aslıcan Karaca, and I'm from the Istanbul Technical University Department of Food Engineering. Thank you very much for the introduction, and your presentation was pretty good. Thanks. I will be presenting you a very short, brief topic on protein extraction from waste and byproducts. So why are proteins important, especially plant-based proteins? They have characteristic properties in terms of nutrition and health. And as we all know, there is an increased global demand for food. And since the plant-based proteins have a relatively lower cost and easier to produce on a sustainable aspect because they have lower environmental impact, they have been pretty important during the last years, and those proteins can be obtained from various sources such as cereals, legumes, oilseeds, nuts, and some ancient grains. Since we have time constraints, I won't go into too much detail. And under the plant proteins, there are different types of proteins, and based on their solubility characteristics, they are classified as albumins, globulins, and prolamins. Those different groups are soluble in different environments. And in case of protein extraction methodologies, especially from wastes and byproducts, there are different methods. Each have advantages and disadvantages. And the most commonly applied methods can be listed as isoelectric precipitation, air classification, salt extraction, ultrafiltration, and enzyme-based methodologies. As I mentioned, each have advantages and disadvantages, and depending on the raw material you use, and also depending on the extraction technology you use, the composition of the protein extract changes. The ratio of globulins, albumins, and prolamins can change, and when the composition of the protein changes, its functionality changes as well. The functional properties of proteins can be listed as solubility, viscosity, emulsifying, foaming, water, fat holding, and gelation properties. And those properties are very important in terms of the performance of the protein in the end product. And they are affected by the protein composition, also environmental factors such as temperature, pH, etc. And as I mentioned, the raw material that is used and also the production method that is used affects the composition of the protein, and it also affects the functionality of the protein. So how do we choose? There are important parameters such as the cost, availability, functionality, taste, labeling claims, and nutritional profile, etc. And the proteins that are extracted can be used in a variety of end products, and the main application areas are snack foods, beverages, and bakery products. Those are the most commonly used categories. And in case of extraction of those proteins from waste and byproducts, certain categories have come to the attention of academia recently. And as I mentioned, the waste and byproducts can be utilized for protein extraction. In case of the cereal group, brands of wheat, buckwheat, and rice are widely utilized for protein extraction. In case of seeds, pressed cakes of oil seeds such as flaxseed, canola, sesame seed, and pumpkin seed cakes are used for protein extraction. Legumes are a big category as well. The pea pods, the hulls, and the milling byproducts are utilized for protein extraction. In case of nuts, the pressed cakes of almond, walnut, and hazelnut are also used for extraction of proteins. And those are examples to the studies on lab scale. Those are from the literature studies. And I want to move on to the challenges because I know I have a very limited amount of time left, I guess, right? The challenges in extraction of protein from waste and byproducts can be listed as increasing protein extraction yield, improving functionality and nutritional quality, and also improving sensory properties of the protein extracted from the waste and byproducts. And in order to overcome these challenges, different strategies can be employed. Some enzymatical methods can be employed, and sometimes novel extraction technologies are utilized as well. And in case of flavor challenges, there are different strategies used as well. And I know I'm approaching the end of my time, so I'm ready if you have any questions or comments. Thank you for listening.
Thank you so much. Yeah, nice work you have presented. I have one question for you. So, is there any underutilized crop, or you have estimated any underutilized species of crop for the protein analysis?
Yeah. If we think about the underutilization issue, this legumes come into attention because the byproducts of legumes are very rich in protein. The husks, the pods, or the processing water, which is also known as aquafaba, they are very rich in water, protein. They can be utilized for protein extraction, but there are some challenges. Protein extraction yield is low. It's difficult to scale up the process. So it is not conducted on an industrial scale, and it needs there has to be some improvements are needed. Yeah.
Thank you. Thank you. Yeah. Next, please.
Next, I would like to invite Sadhana Jadon and Salim Siddiqui, and the title of their presentation is Influence on the Human Body Due to the Relationship Between Phenolic Compounds Intake and the Human Gut Microbiota.
Yes, ma'am, I'm sharing my slides. Is it visible? My slides are visible.
Not yet. Yes, now it is.
Very good afternoon to all present over here. Myself, Sadhana Jadon, and I'm here research scholar in Department of Life Sciences, Harvard University, and I'm working under Professor Salim Siddiqui, sir, School of Agricultural Sciences, Harvard University. So my topic of presentation is influence on human body due to the relationship between phenolic compound intake and human health. So let's understand why it is important to understand the relationship between the phenolic compound and the gut microbiota and what are their influence on the human body. So it is known that polyphenols are basically secondary plant metabolites which constitute a diverse group of compounds. But the main problem related to the polyphenol is that they show the poor bioavailability. Because of their structure or many other factors such as like host-related factors or food processing methods and their interaction with the food methods. But it has been shown that when they interact with the intestinal microorganisms, they are metabolized by the intestinal microorganisms and they are converted into the low molecular weight compounds, which enhances their bioavailability. So in such case, there is an interrelationship between the polyphenolic groups and the gut microbiota, because the polyphenolic group they have the bidirectional relationship because the polyphenolic compound they also act as a prebiotic which enhances
Your slide is still. Your slide is still.
Now, ma'am, is it changed? Hello?
Yes, it is. Now, please continue.
Yes, ma'am. So at the same time, these polyphenolic groups they act as an antibiotic, prebiotic substrate and increase the growth of beneficial bacteria. And at the same time, this gut microbiota it helps in the enhancing the bioavailability of polyphenolic group. So if we talk about the gut microbiota, so all multicellular organisms they live in a strong bond with the microorganisms, and so humans are not exception. So human gut microbiota harbors a complex microbial community that is known as the gut microbiota, and human gut microbiota contain 100 trillion microbes, 3 million genes, and so not just gut, this microbes it colonize almost other parts of the body, but gut is the most colonized organ which almost 70% of the human microbes. And so major phyla in gut include vermicutes about 64%, encompassing genera Lactobacillus, Bacillus, Clostridium, Enterococcus, Luminococcus, bacteria, Calibacterium, and Rosifera. And second phylum includes bacteroides, about 23% comprising bacterioles and Prevotella, and actinobacteria and group microbia is very less in numbers. So there is a variation in gut microbiota because the gut microbiota it differs person to person. Individual to individual, and it is influenced by the various several factors like endogenous factor and the exogenous factor, including endogenous factors including age, genetic, birth, gestational date, mode of delivery, and varying feeding habits, weaning periods, and hormonal changes. Exogenous factors like some diet, dietary pattern, and medication, health conditions, geographical reasons, pollution, labeling. Yes, ma'am.
You have only 1 minute to conclude.
And these gut microbiota, they are very important. They play an important role in supporting the immune and the metabolic functions of the body. Whereas the polyphenolic compounds, they have very good properties, such as antioxidant property, anti-inflammatory properties, cardioprotective properties, immutable properties, including neuroprotective properties. So they act as the antioxidant properties because of their ability to inactivate or prevent the formation of reactive free radicals because of presence of hydroxyl groups in their structure. And so they are considered as a new class of prebiotics because their resistance to host digestion capacity of being fermented by intestinal microorganisms and stimulation of intestinal bacteria. So in such case, uh, they are various recent studies have demonstrated the beneficial effect of polyphenol by stimulating the growth of beneficial bacteria and inhibiting or reducing the growth of pathogenic bacteria. So uh, vegetables such as carrot, broccoli, beetroot, cauliflower, potato, they are good source of polyphenol. And so various studies have been shown that
Adna,
one of the
please come
studies.
to the conclusion. You have already exceeded the time limit.
Just ma'am, just in 1 minute so I can conclude.
Just come to the conclusion, please.
Okay, so according to various studies, it has been shown that it promotes the growth of good bacteria and inhibits the growth of the pathogenic bacteria. So this gut microbiota, it helps in the enhancing the viability of polyphenols by converting into the low molecular weight compounds. So let's jump to the conclusion. So polyphenols. So it has been shown that like polyphenols can be used to modulate the intestinal microbiota as a role added to their main properties like antioxidant and antimicrobial properties. And so in a conclusion, by summarizing the latest studies, highlight the probiotic role of polyphenol. They have the ability to influence both modulation of gut microbiota and food journal health through their beneficial effect by driving from different mechanisms and impacting multiple organs and systems. Thank you.
Okay, thank you. Next, please.
Next, I call Shreya, Sheetal Deshmukh, and N. Tejo Kalyani. And the title of their presentation is Novel Approach of Using LEDs to Grow Lettuce Hydrophonically.
Am I audible? Hello?
Yes, you are audible, Shreya.
Very good morning to all the jury members present here. Myself, Shreya Raghote from Lakshmi Narayan Institute of Technology, studying at third year in food technology. Today, I'm here to present the novel approach of using LEDs to grow indoor lettuce. It is a technological approach towards the food industry or cultivation industry under the guidance of Dr. Sheetal Deshmukh and Dr. N. Tejo Kalyani ma'am. As we all know that the world is going towards focusing towards the healthy diet food, and it is not very possible in every country in every season, but there are lots of foods that is available in the market but not bestowed with all the nutrients needed to us. But there is one vegetable which is available in world, but in not every country because of it is seasonal. That is lettuce. Lettuce is a green leafy vegetable which botanical name is Lactuca sativa, belongs to family Astaceae, which found abundantly in China, Japan and USA, but it is very limited in India. There are different types of lettuce are found in various countries like loose leaf lettuce, romaine lettuce. These are few of them. Iceberg lettuce, which is crunchy one used in salads or soups. Butterhead lettuce. But why it is very limited in India? Because lettuce is winter crop, which need almost 15 to 20 degrees Celsius of temperature and 6 to 8 hours of exposure to sunlight. And India being a tropical country having temperature more than 25 degrees Celsius, it is not possible in India to grow lettuce. But technology can make it possible as like LEDs. But why should we consume lettuce? That it having lots of it is bestowed with lots of properties like antimicrobial properties, antioxidant properties, anti-inflammatory properties, which keep us away from infections, having lots of minerals having lots of minerals which remove toxins from our body. It is rich in fiber, low in calories, so it makes us fat to fit. Also remove various cancer cells from our body, prevent us from cancer, improve insomnia and anxiety, and very good for our heart as it maintain our cholesterol level. There are nutrients like omega-3 fatty acid which build our healthy body. It is rich in vitamins like B6, C, also vitamin A, vitamin K, which strengthen our bone. It is rich in minerals like sodium, magnesium, calcium, potassium. Also, there are zinc, iron. But why it is not very available in India? Because of our climate change. And so therefore we can use novel approach to grow lettuce in tropical countries, that is LEDs. Why LEDs? As it is a non-thermal source of light, it is environmental friendly, energy efficient, affordable, longer lifetime, and also the tunable emission is possible. But along with LEDs, we can also cultivate it hydroponically. Hydroponically is the cultivation where we cultivate the plants using only nutrient solution, which without any soil and sand, gravel, etc. In this process, we add minerals, then add with we take minerals, add with the fresh water and some salt, micronutrients, and make it possible to grow it in the solution and maintain the pH and electric conductivity of that solution. There are different types of LEDs we can use here. We can also use white fluorescent light, white light as it is a combination of all the colors.
Ms. Shreya, you are requested to please wind up your topic. Your time is up.
Okay, ma'am.
Yeah. So within 1 minute, please stand up. Please wind up.
Yes. Role of LED is red LED. It's 620 to 720 nanometer, and it having all this help to growth, anthocyanin accumulation, promote photosynthesis, increasing concentration of phenolic fire synthesis accumulation. Same with blue light, it controls somatofunctioning, simulate photomorphogenesis, anthocyanin biosynthesis, and green light also do the same stretching and chromatal conductance. There are some advantages of LED lighting is that it yields higher number of leaves, healthy appearance, large leaf area, and also it is very similar to naturally grown lettuce as nutrients and antioxidants are retained by the use of
Thank you,
organic
Shreya. Your 1 minute is over. So if there are any queries from the judges side, then they may please ask. Otherwise, we will proceed with the next.
Yeah.
Yes.
No,
If there is any question, I am ready to
actually, we
answer.
are short with the time, so I feel that we need to complete it. Please call next.
Next, we have Shifra Jha and Anupam Roy.
Thank you, ma'am.
Thank you, Shreya.
Next, we have Shipra Jha and Anupam Roy, and the title of their presentation is Adsorption and In Vitro Dissolution Study of Quercetin Loaded into Starch-Based Expanded Porous Matrix, Kinetic and Isotherm Study.
Please start, Ms. Shipra. And please stick to the timeline because the session time is already over and we are running behind. There is next to the valedictory session is there. Shipra? Ms. Shipra Jha? You are not audible.
Hello, yes, ma'am. Am I audible now?
Yes, please.
Today I will be presenting part of my PhD thesis, which is entitled Adsorption and In Vitro Dissolution Study of Quercetin Loaded into Starch-Based Expanded Porous Matrix, Kinetic and Adsorption Studies. So, quercetin is a natural occurring flavonoid. It is generally found in fruits, vegetables such as onion, apple, and tea. It is an excellent antioxidant, anti-inflammatory, and have anti-cancerous activity. It is natural, non-toxic, and non-carcinogenic and harmless product in human diet. The main mechanism responsible for its all activity is its ability to quench the reactive oxygen substance. Thus, it decreases the oxidative stress responsible for the various disease. But with all these advantages, its hydrophobic nature, low adsorption in GI tract,
Ms. Shipra, you just have 3 more minutes. So I would suggest you that you please come to the what you actually want to present.
Okay.
There you will be able to present only 5 to 7 slides.
Okay, ma'am. So with this advantage, it has problem of low solubility. To overcome this, we have loaded this quercetin into starch-based expanded porous matrix, which is produced using STST treatment. This is a cheap source for and replicate porous starch. So the quercetin was loaded into porous starch using fixed bed column. This is the schematic diagram of the column. And then the adsorption studies were done. This is the diagram of quercetin in different form. This is the pure quercetin. This is quercetin loaded in organic solvent, and then when quercetin is loaded into the porous matrix, it has been found that it is successfully loaded into the porous matrix. So then static adsorption and desorption tests were performed, which conclude that porous starch-based porous matrix has good adsorption property for quercetin. Then 2 isotherms, that is Langmuir and Froelich isotherms, were applied to the results obtained, and it was formed based on this result that the adsorption Langmuir as it followed Langmuir monolayer adsorption based on the value of R square, we can see. Then kinetic study, which is pseudo first order and pseudo second order, was done, and it was concluded that based on the result calculated that these results followed pseudo second order reaction kinetics based on the result of R square and QE calculated. And then in vitro resolution was done using USP apparatus. pH of 5.5 was maintained, and from this figure it can be seen that the pure quercetin has very low adsorption, but while quercetin loaded in starch-based expanded porous matrix so slow and sustained release in the pH.
Thank you.
Thank you, ma'am.
Thank you for your presentation, Ms. Shipra, and sticking to the timeline specifically. Any questions from the judges? Okay, then please call the next.
Next, I call Namrata Kolu, and the title of her presentation is Understanding Sustainable Package and Design in Beverage Packaging.
Hi all, good afternoon. When it comes to my presentation, I'll be presenting you the understanding of the sustainable package and design in the beverage industry. This is my topic. I'll quickly take you to the presentation because of the time limit. When it comes to sustainability, globally, people tend to say that we will either go towards a solar energy or we will have the alternative sources of energy to make a package or a product sustainable. But when it comes to the industrial perspective, we need to understand that the emission of the GHG gases becomes the more important initiative for them to achieve the net zero of a sustainable package. So according to the greenhouse gas protocol, there are 3 different scopes that are defined by the GHG protocol. That is the direct emissions that can be controlled by the organization. There are indirect emissions that are controlled by the organization, which are like company facilities and the vendors that are coming in, the purchase, electricity, transportation and distribution. And there is a scope 3 where there are indirect emissions that are not owned and controlled by the organization, like the investments and the employments that are employees that are coming in their own vehicles, which are not sustainable, etc. So here, when it comes to the packaging material, the major scope it comes to is the scope 2. Considering that when it comes to package, when we say it is sustainable, it should be either reuse, recycle, rethink, or reduce. So reuse, recycle, and rethink cannot be globally possible in case of food because of the contamination and recontamination issues. So reduce becomes the more important factor when it comes to the sustainable packaging material in the beverage industry. When it comes to the sustainable packaging material or the basic packaging material that are available in the beverage industry are the returnable glass bottles, there are cans, there are outer cartons, and there are multi-packs and the shrink films for cans. So in this project, what I have worked on is to reduce the weight of the bottle from 410 grams to 390 grams and to study the carbon emission from this. So if you look at this in the diagram, if you can see in the bottom glass, there is always a curve with lots of material has been present. If we are reducing that lots of material which is there at the bottom of the bottle and make it as per the requirements and based on the stability, we can directly reduce the 23 grams of reduction in the carbon, and we can make it the lightest bottles that are possible. Then when it comes to the can also, it's the same. Down gauging of the weight of the cans and the can lids that are present in the factories can also reduce the emissions of the greenhouse gases that are present. So this is a brief presentation and the comparison of like what is happening when we are doing from existing and the proposed ends. And these are like certain communications which clearly says that how much weight reduction has happened when we reduce the gauge of the metal can. So due to the time limit, I'll just quickly take you through. That is on the bottles and cans. The most important factor that is even present in any industry when it comes to the food industry for packaging is outer carton. So down gauging of these outer carton sizes by introduction of the existing is the regular slotted cartons that we have in the market. But introduction of the wraparound cartons will actually reduce 2% of the paper that is being used for the material to form a carton, which is directly moving to 8.9% reduction in the amount of the carbon emissions that are happening through the paper material. Even though it is recyclable, and the packages and the paper is recyclable, and please understand that these still emits carbon. So then there are like the very design packs that we carry, the packs we call them in the market, where you get to carry your alcohols or like beers. Sometimes you can see in the Coke as well. There are like 6 packs, 5 packs that are available to carry in a single pack. These also emits lots of carbon emission because we use paper. Lots of paper is being used in making these cartons. So to reduce the paper for these multi-packs, these are the innovative designs that are coming up globally, which directly impacts and reduces the carbon emission by reduction of the paper that is used. And then we have the shrink films, which are there in the market even for the water and everything. So currently I am looking at reducing the durability and the handling, improving the durability and handling by reducing the amount of the plastic that is basically the poly shrink film. We use the LLDP to reduce the amount that is being used in these packages. So overall, when we come down and see with the can lid down gauging, wraparound carton introduction, round down gauging, totally there is a 55% reduction in the carbon emissions. So this is the conclusion of this project. So we always say that we wanted to go for alternative sources of energy like solar and all. Having a solar plant or a windmill in a plant is very expensive. But if we work towards these small innovations which can be done at the plant, we can directly have a reduction in the carbon emissions from the factory. For example, if you can see, the yellow color one is the GHG protocol globally that is published by the FAO, that is Food and Agriculture Organization. And using the above initiatives that which are discussed, there is a 40% reduction and overall 55% reduction in the emissions. Thank you for giving me this opportunity to present my research work in front of you all, and I'm open for questions.
Thank you so much.
Thank you so much. Next, please.
Next, we have Adil Ansari, and the title of his presentation is Freezing Food and the Factors Affecting Its Stability.
Hello, am I audible?
Yes, you are audible, Adil.
Can you see my screen?
No, it's still not visible. Yeah, now it's visible.
Okay. So good morning, everyone. I am Adil Jamal from Dr. Oker India Private Limited. So I'm working within the cold chain industry, and I thought to give a little theoretical contribution to the food industry by explaining some methods of frozen technology. So due to the time limit, let's come straight to the methods and the outcomes. So so this research is basically based on the effects of freezing and frozen storage on the food quality and the factors influencing the properties of frozen meals. So in this research, I've taken a sample of meat, which is lamb loin chops, and cut it into three shapes, which is cube, cuboid, and cylindrical. So we have taken 9 pieces of samples in which we have kept 3 pieces as control samples inside the chillers. 3 pieces as samples we have kept inside the deep freezer at minus 15 degree, and 3 pieces inside the air blast freezer, which is at minus 35 degrees Celsius. So after 24 hours, we have taken out the samples and thawed it for 6 hours in an incubator. Then we have determined the drip loss, and in the second experiment, I have calculated the weight losses with respect to the different freezing rates. So these are the results that we have got. So in table 1, I have shown the drip loss values using the different freezing methods. The results clearly states that the mean values of drip loss of the different meat cuts was around 9 to 10%, though the control samples stored in the chiller at 2 degrees Celsius showed a mean drip loss of 4 to 5%. So this concludes that the drip loss of the thawed samples was found to be higher than the controlled ones. And these results are also in agreement with the Annan experiment for examining the meat quality. They also found out that no major differences were observed using different freezing temperatures. And it was also discovered that apart from the freezing conditions, the quality of frozen meats also depends upon the various post-mortem treatments, such as loss of ATP energy and the reduction in levels of pH. So to conclude my research, so we have discovered that the drip loss of the thawed samples was found to be higher than the control samples. However, it was found out that no significant differences were observed in the mean values of drip loss using different freezing temperatures. Furthermore, it was discovered that slowly frozen meat lost more weight during freezing and thawing than the fast frozen meat. And as stated in many studies earlier, freezing has a little impact on the quality and nutrition of the food, but we can improve by the investment in the frozen technology. For instance, if high power and ultrasound assisted freezing will be made economically feasible, then it can be used in the industry for the better process results. Thank you.
Okay, thank you so much. Next, please.
Thank you, Adil,
Thank you, ma'am.
for sharing your information. Thank you. Can we have next, please?
Yes, ma'am. Next, we have Muskan Khan, and the title of her presentation is Calcium Fortified Bread.
Good afternoon, everyone. Am I audible?
Yes, please. And please stick to the timeline of 4 minutes.
Yes, ma'am, for sure.
Just try to turn on your camera so that we can see you.
Yes, ma'am, for sure. Ma'am, is my screen is visible?
Yes, it's visible.
Okay, am I visible too?
Yes, you please continue.
Okay, so good afternoon to everyone present here. I am Muskan Khan, student of Shahid Rajguru College. Is glad to get this opportunity to present my research work in front of you all. So the topic of my paper representation is calcium fortified bread. This research work was carried out by me along with my colleague Arshi under the guidance of Ms. Prabjot Kaur, the assistant professor of our college. The main focus of our work was to develop an innovative food product with nutritional properties as well as by utilizing the biodegradable waste. For sustainable development, waste should be recycled and reused. This aids in protecting the environment and yield value-added products during zero waste standards. Eggs are consumed on a regular basis by common man and children all around the world.
Muskan, do you have proper PPT with you? Because it's since I think you are sharing from your phone, nothing
Yes,
is
ma'am.
visible to us.
Yes, ma'am. No, ma'am. Actually, I don't have PPT right now in my phone. Even I asked, they said that we can present our PDF paper also.
You can present your PDF paper, but it has to be in a PowerPoint presentation only, Muskan. That is the rule of the oral presentation.
Okay, ma'am. But I didn't have
You
it right.
please directly come to your conclusions.
Okay, ma'am. I'll directly come to my conclusion.
Yeah.
Now is it visible, ma'am?
It will not be visible because on your phone the PPT, the full paper length doesn't appear properly. So it will not come. You just summarize your
Okay, I'm just
conclusions.
summarizing it. Okay. Now we talk about calcium importance for human. The calcium is a vital element for growth and activity, maintenance of human body. Now, for the incorporation of actual benefits, we thought of bread. It would be a suitable vehicle for the fortification because it is a common part of diet worldwide. Therefore, we came up with the idea of calcium fortified bread, which was fortified using eggshell powder.
Muskan, please come to your conclusion.
Okay, ma'am. So, okay.
No.
The conclusion is,
yes,
ma'am, we have prepared 4 different samples, which was subjected to sensory evaluation. And in that sensory evaluation revealed that sample A with 0.5% eggshell powder was liked the most and accepted among all the 4 samples, which resembles the control sample. Therefore, 0.5% eggshell powder is appropriate for incorporating it in bread. The result sensory evaluation is illustrated in table 3. Okay, but it's not visible. Okay, and we had also conducted a shelf life study of these samples. So whether there is an effect of ESP on bread's shelf life, it is clear that in both the conditions, that is room temperature and refrigeration, control sample and ESP samples behaved in same manner. Therefore, it could be inferred that eggshell powder has no effect on bread's shelf life. By this, I would conclude my paper. Thank you.
Thank you, Muskan. Any questions from the judges?
Thank you so much, ma'am. It's
okay, ma'am. Ma'am, with the permission of yourself and Dr. Deepti, can we conclude the session for today?
Yes, ma'am, please.
More student is
Yes, ma'am.
one more student is there.
Okay.
And that Dr. Vandana, she has not present. I guess.
Ma'am, she is not present. She hasn't come.
Okay. So
One
we have to drop
student is
that now.
Zubia Islam.
Ma'am?
Zubia Islam and Saleen Siddiqui.
Ma'am, they were in between somewhere?
Yes, ma'am, they are newly added in the list.
Okay.
So are these students are present? Zubia Islam and Saleen Siddiqui. Okay, I think
Yes, ma'am. Yes, ma'am.
Okay.
Give me 2 minutes, ma'am. Okay. Is it visible?
Yes, it's visible, Zubia.
Hello.
Yes, it's visible.
Hello.
And Zubia, yeah, you have to wind up within next 3 minutes. So please start with your presentation, Zubia.
Okay. Good afternoon, everyone. Myself, Zubia Islam, PhD scholar from Sharda University. And today I am going to present my topic, modified starch, an ingredient possessing value-added attribute for innumerable application in food industry.
Yes, you are not audible.
Zubia, you
Start with introduction. Starch is a polysaccharide found in.
Zubia,
Sorry.
can you please skip the introduction? We have already a limited.
Sorry, ma'am.
Can
You
please
just
skip the
have,
introduction?
ma'am
Just
is saying, please skip the introduction. You have only 2 minutes to present your slides.
Okay.
Yeah, because
So,
after that we have to end the
modified
session.
starch.
Yeah, please.
Okay, ma'am. Okay. Why starches are modified? Starches are modified to enhance their performance and properties in different applications. Starch may be modified to increase their stability against excessive heat, acid and shear, time cooling, freezing, to change their texture, to decrease or increase their viscosity, to lengthen or shorten gelatinization time, or to increase their viscous stability. Some commercial sources for starch is cereal grains, seeds like maize, wheat, rice, and sorghum. Root and tubers, example potato, sweet potato, tapioca, or cassava, or arrowroot. And then a third one is stem or and pith like sagu. Three methods for starch modification. First is enzymatic, next is chemical method, and third one is physical method. Some recent research at national and international levels are like Viswas et al. Use makhana to extract and modified starch using amylopumilase enzyme, and they found that decreased water holding capacity, oil holding capacity, swelling power with increased cohesiveness, springiness, and gumminess, and increase in resistance starch. Surinder and et al. In 2016 use potato starch and modified the starch by using citric acid and found that starch starch exhibit higher water solubility index and water absorption index and lower gel strength. Third one is Singh et al. 2017 using amaranth flour and modified starch using heat moisture treatment, and they found improved the
Zubia, you
amaranth
are
starch
only left
properties
with
to be
one more
used
minute.
for noodle
Zubia,
production.
you are left with only one more minute, please. So please try to wind up with your conclusions.
Okay, ma'am. Okay. Next one is Zambelli et al. In 2018, starch modification done with cassava and used acid treatment, and they found reduced in apparent viscosity. Now moving on, another slide. Modified starch in market. Functions are thickness, stabilizer, and the types of modified starch methods are three methods like chemical modification, enzymatic modification, and physical modification. Applications are in food and beverages like confectionery, beverage, processed food. Next is animal feed like swine food, ruminant food, poultry foods, and in paper making, weaving, textile, pharmaceutical, and plastic industries. Food containing modified food starch like chips, in soups, instant pudding, low-fat ice cream, cheese sauce, mozzarella, powder-coated food such as cocoa dusted almonds, candy capsules that contain more some medications. Next is
Thank
conclusion.
you, Zubia. We will not be able to give more time. Yeah, you just conclude it. It's already, your time is already up.
Okay, ma'am. Starch continues to be an important renewable biopolymer in both the food and non-food industry. Its property very depend on the plant origin. Starch is rarely consumed in its native form due to the restricted solubility in water, heat resistant gelatinization, and etc., which limits the industrial application. Different modifying procedure could significantly enhance their properties of raw starch by modifying their physicochemical attributes, leading to an increase in the industrial value of the starch. With the world focused on moving towards sustainability, starch has found itself as one of the forefront runner in the development of functional material for advanced application. This is the reference. Thank you.
Thank you so much.
So thank you. Dr. Loveleen, is there any more candidate updated to you? No. So, okay. So we would conclude the session. Akansha, you may please convey the vote of thanks.
Yes, ma'am. A very good afternoon to everyone. On behalf of all the students and staff, and I'm glad to be standing here to express my vote of thanks. I would like to extend my heartfelt gratitude to our esteemed guests, Dr. Sujata Pandit Sharma, Assistant Professor, Department of Life Sciences, Sharda University, and Dr. Deepti Sharma, Assistant Professor, Shyama Prasad Mukherjee College for Women, University of Delhi. Sincere thanks to the coordinators of the event, Dr. Neha Sharma and Dr. Loveleen Sharma, who handled the event throughout. A wide round of applause and thanks to all the participants who made the event a memorable one. Finally, I would like to thank all of you present here for making the time to be with us today and helping us make this event a grand success. Thank you, everyone, and I request everyone to join the valedictory session at 2:30 p.m. through the link that has already been provided to you. And all the students from Amity are requested to move to the moot court for the valedictory session.
So thank you. Thank you, Sujata ma'am. Thank you, Deepti ma'am.
Thank you,
With this,
ma'am.
Thank you, ma'am. Thanks, ma'am.
thank you.